Hello there, and welcome to this quiet space where time slows down and the history of our planet can unfold without hurry. Tonight, we're going to step back, not centuries or millennia, but so far into the past that the Earth itself becomes almost unrecognizable. We're traveling to a period that lasted longer than most minds can comfortably hold, 1 billion years. Not 1 million, not 100 million, a full billion years of planetary time, stretching from 1.8 billion years ago to 800 million years ago. This era earned itself an unusual name among scientists. They call it the Boring Billion. The name sounds almost dismissive, as if nothing of consequence happened during this vast stretch of deep time. And in a sense, that's exactly what the name suggests. For nearly 1 billion years, the Earth seemed to pause. The continents barely moved. The climate remained stable. The oceans held their chemical composition with little variation. And life, which had been evolving at a rapid pace before this period and would explode into complexity afterward, appeared to simply stop advancing. But the story of the Boring Billion is not really about boredom at all. It's about constraint, about pressure applied slowly over unimaginable spans of time, about how a planet can trap its own biology in a chemical cage and force evolution to work beneath the surface, hidden from view, preparing for changes that wouldn't arrive for hundreds of millions of years. Before we begin, I'd be grateful to know where you're listening from tonight and what time it is in your corner of the world. It's a strange thing to think about. Here we are, scattered across the globe, separated by miles and hours, yet all of us looking back together toward the same ancient moment. If you find this kind of slow, careful exploration of deep time worth your attention, a like or subscription helps this reach others who might be searching for the same kind of quiet curiosity. No pressure, just an invitation. So, settle in. Let the day fall away. We're going to descend into a period of Earth's history that demanded patience from life itself, a time when the planet taught its organisms to wait, to refine, to prepare, and we'll move through it the way it moved, slowly, steadily, with the calm rhythm of geology itself. There are moments in Earth's history that announce themselves with violence, volcanic eruptions that darken the sky for years, asteroid impacts that reshape continents, ice sheets that swallow entire hemispheres. These are the events that leave clear marks in the geological record, boundaries that scientists can point to and say with confidence, here, everything changed. The Boring Billion is not one of those moments. It is instead the opposite. A vast expanse of time defined not by what happened, but by what didn't. From roughly 1.8 billion years ago to 800 million years ago, the Earth entered a state of profound stability. The kind of stability that, paradoxically, becomes its own form of constraint. To understand this period, you first need to see where it sits in the larger timeline of planetary history. The Boring Billion occupies the middle chapters of what geologists call the Proterozoic Eon. More specifically, it aligns almost perfectly with the Mesoproterozoic era, which formally runs from 1.6 billion to 1 billion years ago. But the informal boundaries stretch a bit wider, encompassing the tail end of the Paleoproterozoic and the early stages of the Neoproterozoic. What matters is not the exact dates, but what came before and what came after, because the Boring Billion sits between two of the most dramatic transformations in Earth's history. On one side, in the deep past, lies the Great Oxygenation Event, an environmental catastrophe that fundamentally altered the chemistry of the atmosphere and oceans. On the other side, in the future, waits the Neoproterozoic, a time of extreme planetary instability marked by episodes of global freezing so severe that ice may have covered the entire surface of the Earth. The Boring Billion is the quiet interval between these storms, 1 billion years of equilibrium, and during that equilibrium evolution appeared to stall. When scientists examine the rock record from this time, they find something unusual, a lack of change. The types of fossils don't vary much. The chemical signatures in ancient sediments remain consistent. The indicators of climate, ocean chemistry, and atmospheric composition all suggest a planet locked in a steady state. For a long time, researchers interpreted this as a failure of life to advance, a period where biology simply treaded water, producing nothing new, achieving nothing remarkable. The name itself reflects that early interpretation: boring, uneventful, a gap in the story. But that interpretation has shifted, because when you look more closely at what was happening during this billion-year span, you start to see something different. Not stasis, but preparation. Not failure, but forced innovation under extreme constraint. The boring billion wasn't a time when evolution stopped. It was a time when evolution had to work differently, slowly, invisibly, building the foundations for everything that would come later. The planet itself was unusually calm during these years. There is no geological evidence of glaciation anywhere in the rock record from this period. No ice sheets, no widespread cooling events. The climate remained temperate and stable across hundreds of millions of years. The continents, which we'll explore in more detail shortly, were assembled into supercontinents that persisted with minimal movement. Mountain ranges eroded slowly. Volcanic activity remained subdued. The tectonic engines that drive planetary change were running, but at a crawl. This stability extended into the oceans as well. The marine carbon cycle, which measures how carbon moves between the atmosphere, oceans and rock, showed little variation. The chemical balance of seawater remained largely unchanged. There were no major shifts in ocean acidity, no dramatic changes in salinity, no widespread turnovers in deep-water circulation. The oceans, like the continents, simply persisted, and yet, beneath this apparent calm, the planetary system was imposing limits. Limits that life could not overcome through sheer evolutionary speed. The stability that defined the boring billion wasn't benign. It was restrictive. A planet in equilibrium is a planet where resources cycle slowly, where nutrients remain locked away, where opportunities for rapid change are few. Life, during this time, had to adapt not to chaos, but to scarcity. To a world that provided just enough to survive, but rarely enough to thrive. And over the course of 1 billion years, that constraint became a kind of teacher. It forced organisms to become more efficient, to develop new strategies for managing limited resources, to build complexity not through rapid diversification, but through slow, patient refinement of cellular machinery. The Great Pause, then, is not really about boredom. It's about pressure. The kind of pressure that comes not from sudden disasters, but from a planet that refuses to provide what life needs to grow. And as we'll see, that pressure had profound consequences. Not immediately, not dramatically, but slowly, steadily, over spans of time almost too long to imagine. The boring billion was the crucible, the quiet forge where the tools for complex life were made. And to understand how that happened, we need to look at what made this period so uniquely constraining. Starting with what came before, there are moments in Earth's history that announce themselves with violence. Volcanic eruptions that darken the sky for years. Asteroid impacts that reshape continents. Ice sheets that swallow entire hemispheres. These are the events that leave clear marks in the geological record, boundaries that scientists can point to and say with confidence, here, everything changed. The boring billion is not one of those moments. It is instead the opposite. A vast expanse of time defined not by what happened, but by what didn't. From roughly 1.8 billion years ago to 800 million years ago, the Earth entered a state of profound stability, the kind of stability that paradoxically becomes its own form of constraint. To understand this period, you first need to see where it sits in the larger timeline of planetary history. The boring billion occupies the middle chapters of what geologists call the Proterozoic Eon. More specifically, it aligns almost perfectly with the Mesoproterozoic era, which formally runs from 1.6 billion to 1 billion years ago. But the informal boundaries stretch a bit wider, encompassing the tail end of the Paleoproterozoic and the early stages of the Neoproterozoic. What matters is not the exact dates, but what came before and what came after. Because the boring billion sits between two of the most dramatic transformations in Earth's history. On one side, in the deep past, lies the Great Oxygenation Event, an environmental catastrophe that fundamentally altered the chemistry of the atmosphere and oceans. On the other side, in the future, waits the Neoproterozoic, a time of extreme planetary instability marked by episodes of global freezing so severe that ice may have covered the entire surface of the Earth. The boring billion is the quiet interval between these storms, a billion years of equilibrium. And during that equilibrium, evolution appeared to stall. When scientists examine the rock record from this time, they find something unusual, a lack of change. The types of fossils don't vary much. The chemical signatures in ancient sediments remain consistent. The indicators of climate, ocean chemistry, and atmospheric composition all suggest a planet locked in a steady state. For a long time, researchers interpreted this as a failure of life to advance, a period where biology simply treaded water, producing nothing new, achieving nothing remarkable. The name itself reflects that early interpretation: boring, uneventful, a gap in the story. But that interpretation has shifted, because when you look more closely at what was happening during this billion-year span, you start to see something different. Not stasis, but preparation. Not failure, but forced innovation under extreme constraint. The boring billion wasn't a time when evolution stopped. It was a time when evolution had to work differently, slowly, invisibly, building the foundations for everything that would come later. The planet itself was unusually calm during these years. There is no geological evidence of glaciation anywhere in the rock record from this period. No ice sheets, no widespread cooling events. The climate remained temperate and stable across hundreds of millions of years. The continents, which we'll explore in more detail shortly, were assembled into supercontinents that persisted with minimal movement. Mountain ranges eroded slowly. Volcanic activity remained subdued. The tectonic engines that drive planetary change were running, but at a crawl. This stability extended into the oceans as well. The marine carbon cycle, which measures how carbon moves between the atmosphere, oceans and rock, showed little variation. The chemical balance of seawater remained largely unchanged. There were no major shifts in ocean acidity, no dramatic changes in salinity, no widespread turnovers in deep water circulation. The oceans, like the continents, simply persisted. And yet, beneath this apparent calm, the planetary system was imposing limits. Limits that life could not overcome through sheer evolutionary speed. The stability that defined the boring billion wasn't benign. It was restrictive. A planet in equilibrium is a planet where resources cycle slowly, where nutrients remain locked away, where opportunities for rapid change are few. Life during this time had to adapt not to chaos, but to scarcity, to a world that provided just enough to survive, but rarely enough to thrive. And over the course of a billion years, that constraint became a kind of teacher. It forced organisms to become more efficient, to develop new strategies for managing limited resources, to build complexity not through rapid diversification, but through slow, patient refinement of cellular machinery. The Great Pause, then, is not really about boredom. It's about pressure. The kind of pressure that comes not from sudden disasters, but from a planet that refuses to provide what life needs to grow. And as we'll see, that pressure had profound consequences. Not immediately, not dramatically, but slowly, steadily over spans of time almost too long to imagine. The boring billion was the crucible, the quiet forge where the tools for complex life were made. And to understand how that happened, we need to look at what made this period so uniquely constraining. Starting with what came before. During the boring billion, Earth's continents were not scattered across the globe as they are today. Instead, nearly all of the planet's landmass was locked together in vast supercontinents that persisted with little movement for hundreds of millions of years. These continental configurations played a central role in creating the environmental conditions that defined this era. The first of these supercontinents was called Columbia, though some researchers refer to it as Nuna. It began to assemble around 2 billion years ago as smaller continental fragments collided and fused together along massive mountain ranges. By one point, 7 billion years ago, Columbia had reached its maximum extent, stretching across a large portion of the planet's surface. Reconstructing the exact shape and position of Columbia is difficult. The rocks that would provide clear evidence have been deformed, buried, and in many cases eroded away over the intervening time. But paleomagnetic data, which records the orientation of Earth's magnetic field frozen into ancient rocks, suggests that Columbia was positioned primarily in equatorial and low-latitude regions. This meant the supercontinent sat in the warmest part of the planet's climate system, bathed in direct sunlight year-round. The assembly of a supercontinent is normally a time of intense geological activity. Continental collisions create mountain ranges. Volcanic arcs form along subduction zones. Earthquakes reshape the landscape. But once a supercontinent is fully assembled, something different happens. The tectonic activity slows. The mountain ranges stop growing and begin to erode. The volcanic systems quiet down. The continent enters a phase of stability. This is what happened with Columbia. For hundreds of millions of years, from roughly 1.7 billion to 1.3 billion years ago, the supercontinent persisted with minimal change. There were no major breakup events, no significant rifting. The tectonic engines that drive continental drift continued to operate, but they ran slowly, grinding away beneath a stable surface. This stability had direct consequences for the global environment. When continents collide, they push up mountains. Mountains, in turn, create high topography, and high topography means increased erosion. Rain falls on steep slopes, washes down mountainsides, and carries dissolved minerals into rivers and eventually into the ocean. This process, called chemical weathering, is the primary mechanism by which nutrients are delivered from land to sea. But during the Boring Billion, topography was low. The mountain ranges that had formed during Columbia's assembly had eroded down. There were no new mountains being pushed up to replace them. The continents became flat, stable platforms, and flat continents erode slowly. The rivers that drained these landscapes carried less sediment, fewer dissolved minerals, and critically, fewer nutrients into the ocean. This created a nutrient bottleneck. The oceans, which depend on continental weathering to supply essential elements like phosphorus and trace metals, were starved. The input of these nutrients dropped to extremely low levels. And because life in the oceans depends on these nutrients, the entire marine biosphere was constrained by this scarcity. Around 1.3 billion years ago, Columbia began to fragment. The exact mechanisms are still debated, but the evidence suggests that the supercontinent broke apart relatively slowly, splitting into smaller continental blocks that drifted apart over millions of years. This fragmentation continued for perhaps 200 million years, until around 1.1 billion years ago, when a new supercontinent began to assemble. This second supercontinent is called Readiness. Its assembly brought the continental fragments back together, though in a different configuration than Columbia. Readiness was also positioned in low latitudes, maintaining the equatorial concentration of landmass that had characterized the earlier supercontinent. And like Columbia, Readiness persisted in a state of tectonic stability for hundreds of millions of years. The importance of this continental configuration can't be overstated. By concentrating all the planet's landmass in the equatorial zone, the supercontinents created a climate system that was unusually uniform. There were no polar continents to accumulate ice, no high-latitude landmasses to create temperature gradients. The climate remained warm and stable, with little seasonal variation. But this uniformity came at a cost. A planet with stable continents is a planet with slow geological cycles. The weathering rates that supply nutrients to the ocean decrease. The volcanic activity that releases carbon dioxide and other gases into the atmosphere diminishes. The tectonic recycling that brings fresh minerals to the surface slows down. Everything that drives planetary change operates at reduced speed. And for 1 billion years, this is how Earth existed. 2 successive supercontinents, each positioned in the warmest part of the planet, each persisting for hundreds of millions of years with minimal change. The continents were flat, the climate was stable, and the geological processes that might have disrupted this equilibrium simply didn't occur. This tectonic stillness was one of the defining features of the Boring Billion. It wasn't just that the climate was stable, or that oxygen levels plateaued. It was that the continents themselves had stopped moving in ways that would have altered the global environment. The planet had entered a phase where the very foundation of geological activity was suppressed, and life, which had been evolving rapidly in the chaotic environments of the Paleoproterozoic, now faced something entirely different. Not a crisis, not a disaster, but a world where change itself had become rare, a world where the continents barely moved, the climate barely varied, and the opportunities for adaptation were few. The supercontinents, in other words, created the frame within which the boring billion unfolded. They were the tectonic foundation of the Great Pause, and their persistence, their stubborn refusal to break apart and redistribute, ensured that the constraints imposed on life would last not for millions of years, but for a full billion. Climate during the boring billion was stable in ways that modern Earth never experiences. The planet had no ice ages, no dramatic swings in temperature, no periods of extreme heat or cold. For a billion years, the global climate remained locked in a narrow, temperate range. This stability, which might seem ideal for life, turned out to be one of the most powerful constraints on biological evolution. To understand why, you need to consider what climate stability actually means. On modern Earth, climate varies constantly. Ice ages come and go on 100,000-year cycles. Sea levels rise and fall. Ocean currents shift. Monsoon patterns change. This variation creates ecological pressure. Species adapt to new conditions, migrate to new habitats, or go extinct. And in response to this pressure, evolution accelerates. But during the boring billion, this variation simply didn't exist. The geological record shows no evidence of glaciation anywhere on the planet during this time. No ice sheets formed at the poles. No glacial deposits appear in the rock record. The polar regions may have been cool, but they weren't cold enough to accumulate permanent ice, and the equatorial regions, where the supercontinents were positioned, remained warm and stable. This lack of glaciation is unusual. Earth's history is punctuated by ice ages. They occur roughly every few 100 million years, sometimes more frequently. The Huronian glaciation, which preceded the boring billion, was one of the longest and most severe. The Cryogenian glaciations which followed it may have frozen the entire planet. But in between these two extremes lies a billion-year gap with no ice. The reason for this stability is tied to the position and configuration of the continents. Ice sheets form when continents are positioned at high latitudes, where temperatures are low enough for snow to accumulate year after year. But during the boring billion, the supercontinents were positioned in equatorial regions. There was no landmass at the poles to support ice sheet growth. And without ice sheets, the climate feedback mechanisms that amplify cooling and drive glaciation never engaged. At the same time, atmospheric carbon dioxide levels appear to have remained relatively stable. Volcanic outgassing, which releases carbon dioxide into the atmosphere, was subdued because tectonic activity was low. But weathering, which removes carbon dioxide from the atmosphere by reacting it with minerals on land, was also slow because the continents were flat and erosion rates were low. These two processes balanced each other, maintaining atmospheric carbon dioxide at levels that produced a temperate climate without extreme variations. Ocean temperatures also remained stable. The deep ocean appears to have been warm, possibly warmer than it is today. There's no evidence of the kind of temperature stratification that occurs in modern oceans, where cold polar water sinks and flows along the ocean floor toward the equator. Instead, the boring billion oceans may have been relatively uniform in temperature, with warm water extending even into the deep basins. This thermal uniformity had consequences. In modern oceans, temperature differences drive circulation. Cold water sinks at the poles, flows along the ocean floor, and eventually upwells in other regions, bringing nutrients from the deep ocean to the surface. This circulation is critical for marine life. It delivers nutrients to sunlit surface waters, where photosynthetic organisms can use them. But in the boring billion oceans, with less temperature difference between surface and deep water, this circulation was weaker. Nutrients that settled into the deep ocean tended to stay there. The deep water became stagnant, cut off from the surface, and without vigorous circulation to bring nutrients back up, the surface waters where photosynthetic life thrived remained nutrient poor. The climate stability also meant that rainfall patterns were consistent. There were no dramatic monsoons, no shifting rain belts, no alternating wet and dry periods that might have driven pulses of nutrients from land to sea. Rivers flowed steadily, but without the seasonal flooding or storm-driven erosion that delivers large amounts of sediment and dissolved minerals to the ocean. The nutrient flux from land remained low and constant. Seasonal variation, which on modern Earth drives much of the rhythm of life, was minimal. With the continents positioned in equatorial regions, the amount of sunlight they received varied little throughout the year. There were no strong seasonal temperature swings, no winter freezes, no summer droughts. Life existed in a world where one day was much like the next, one year much like the last, for millions upon millions of years. This might sound like paradise, a planet without extremes where conditions were always moderate, always predictable. But from an evolutionary perspective, it was a trap. Evolution accelerates under pressure. When environments change, organisms must adapt or die. When resources become scarce in one region but abundant in another, migration and diversification follow. When seasons shift, reproductive strategies evolve. But in a world without seasons, without climate variation, without dramatic environmental change, that pressure disappears. There's no advantage to adapting to cold because cold never comes. There's no advantage to developing drought resistance because droughts don't occur. The evolutionary incentive to change, to experiment, to diversify, simply isn't there. And so, life persisted, but it didn't advance. The microbial communities that dominated the oceans remained largely unchanged. The organisms that thrived in the stable, warm waters of the boring billion were those that had already adapted to those conditions. There was no reason for them to become something different, no selective pressure pushing them toward greater complexity. The climate, in its relentless stability, became a cage. Not a hostile environment that killed, not a chaotic system that forced adaptation, but a perfectly moderate, perfectly predictable world that offered no reason to change. And for 1 billion years, life remained trapped in that cage, unable to break free, waiting for the planet to offer something different. Continental weathering is one of Earth's most fundamental processes. Rain falls on rock. The water, slightly acidic from dissolved carbon dioxide, reacts with minerals in the rock surface. Chemical bonds break, elements dissolve. The water carries them away, down slopes, into streams, into rivers, and eventually into the ocean. This process is how nutrients move from land to sea. It's how the ocean gets the phosphorus, iron, and trace metals that life requires. During the boring billion, this process nearly stopped. Not entirely, but close enough that the marine biosphere was starved of essential nutrients for hundreds of millions of years. The reason was simple. The continents were flat, and flat continents erode slowly. When mountains rise, they create steep slopes. Water flows quickly down those slopes, cutting channels, carving valleys, breaking rock apart through physical and chemical weathering. High mountains also create rain. As moist air is forced upward over mountain ranges, it cools, and the moisture condenses into heavy rainfall. This rain accelerates weathering. The combination of steep slopes and abundant water means that young, high mountain ranges weather rapidly, delivering large amounts of dissolved minerals to the ocean. But during the boring billion, there were no young mountains. The mountain ranges that had formed during the assembly of Columbia had long since eroded down. What remained were low, rolling hills and vast, flat plains. Water moved slowly across these landscapes. Rivers meandered rather than rushed. The chemical reactions that dissolve minerals proceeded at reduced rates because the rock surfaces weren't being broken up and exposed as quickly. Tectonic stability compounded this problem. New mountains form when tectonic forces push continents together, or when volcanic activity builds new land. But during the boring billion, tectonic activity was subdued. The supercontinents persisted without major deformation. There were no large-scale continental collisions. No new mountain ranges were being pushed up to replace the old, eroded ones. The continents simply sat, stable and flat, for hundreds of millions of years. This had a direct impact on nutrient delivery to the ocean. Phosphorus, which is essential for DNA, RNA, and cell membranes, comes primarily from the weathering of continental rocks. During periods of high tectonic activity and mountain building, phosphorus delivery to the ocean increases. During periods of tectonic quiescence, it decreases. The boring billion was a time of extreme quiescence. Trace metals followed a similar pattern. Elements like molybdenum, zinc, copper and selenium are required in small amounts by all living organisms. They act as cofactors for enzymes, enabling critical biochemical reactions. These elements are also sourced primarily from continental weathering, and like phosphorus, their delivery to the ocean was severely reduced during the boring billion. The geological evidence for this nutrient scarcity is found in the chemical composition of ancient marine sediments. Rocks formed from ocean sediments during the early part of the boring billion, roughly from 1.8 billion to 1.4 billion years ago, show remarkably low concentrations of bioessential trace elements. The ocean, quite simply, was depleted. This depletion wasn't uniform across all ocean basins or all depths, but the overall trend is clear. The slow weathering rates meant that nutrients entered the ocean at a trickle rather than a flood. And because marine organisms, particularly photosynthetic microbes, depend on these nutrients, the total biological productivity of the ocean was constrained. Productivity, in this context, refers to the rate at which organisms produce biomass. Photosynthetic organisms take carbon dioxide, water and nutrients, and use sunlight to build organic molecules. These organisms form the base of the marine food web. Everything else depends on them. But if the nutrients they need are scarce, their growth is limited. And if their growth is limited, the entire ecosystem is limited. During the boring billion, marine productivity appears to have been low. Not zero, but significantly lower than it would be in later periods when nutrient delivery increased. The oceans were not empty, but they were sparse. Microbial life persisted, but it couldn't expand into more complex forms because the resources needed to build larger, more complex organisms simply weren't available in sufficient quantities. There's a concept in ecology called limiting factors. The idea is that the growth of a population is constrained by whichever resource is scarcest. It doesn't matter if you have abundant sunlight and water if you don't have phosphorus. The lack of phosphorus becomes the bottleneck. During the boring billion, multiple nutrients were scarce simultaneously, creating a situation where life was constrained on multiple fronts. Some researchers have proposed that the preceding biological boom, the one that initiated the Great Oxygenation event, may have consumed much of the planet's readily available phosphorus. Photosynthetic bacteria, thriving in the nutrient-rich oceans of the Paleo-Proterozoic, incorporated phosphorus into their cells in massive quantities. When they died, their remains sank to the ocean floor, taking that phosphorus with them into the sediments where it was buried and removed from circulation. This created a phosphorus debt. The oceans had been stripped of easily accessible phosphorus, and the slow weathering rates of the boring billion weren't sufficient to replenish it quickly. The result was a prolonged phosphorus famine that lasted for hundreds of millions of years. And without phosphorus, complex life simply couldn't develop. The slow erosion, then, was not just a geological phenomenon. It was an ecological constraint, a planetary-scale bottleneck that limited the resources available to life and forced evolution into a narrow channel. Organisms had to become extraordinarily efficient, extracting every possible bit of nutrient from their environment, just to survive. There was no surplus, no excess resources that might have allowed for experimentation or diversification. And so, life persisted in a state of scarcity, not thriving, but enduring, waiting, in a sense, for the planet to provide what it needed to move forward. But the planet, locked in its tectonic stillness, offered nothing more. The erosion continued, slow and steady, delivering nutrients at a rate just sufficient to maintain life, but never enough to allow it to flourish. Oxygen is often thought of as the breath of life. The molecule that powers complex organisms, enables active metabolism, and makes possible everything from flight to thought. But during the boring billion, oxygen was scarce. The atmosphere held only a fraction of what it contains today. And this scarcity imposed one of the most fundamental constraints on the evolution of complex life. To understand this constraint, you need to understand Earth's oxygen history. Geochemists divide it into stages, each representing a different phase in how oxygen accumulated in the atmosphere and oceans. The Great Oxygenation event, which we discussed earlier, represents the transition from stage 1 to stage 2. Before the GOE, atmospheric oxygen was essentially absent. After it, oxygen was present but at very low levels. The boring billion corresponds almost exactly with stage 3 of oxygen history, spanning from roughly 1.85 billion years ago to 850 million years ago. During this stage, oxygen was being produced biologically, but it wasn't accumulating in the atmosphere. The total amount of oxygen in the air remained stuck at persistently low levels, estimated at somewhere between 0.1% and 1% of present atmospheric levels. That's extraordinarily low. Modern Earth's atmosphere is about 21% oxygen. Even 1% would represent less than 5% of current levels. At 0.1%, you're talking about oxygen concentrations that would be immediately fatal to any animal living today. Humans require oxygen concentrations of at least 16% to remain conscious. Below that, unconsciousness occurs within minutes, but the constraint wasn't just about breathing, it was about energy. Oxygen enables aerobic respiration, a metabolic process that extracts far more energy from organic molecules than any anaerobic alternative. A cell using aerobic respiration can produce roughly 18 times more energy from the same amount of glucose than a cell relying on fermentation. This energy surplus is what makes complex multicellular life possible. Large organisms need large amounts of energy. They have tissues to maintain, organs to power, and in the case of animals, muscles to move. All of this requires a steady supply of energy, which means a steady supply of oxygen. But if atmospheric oxygen is at 1% or less, there simply isn't enough available to support large, active organisms. The energy budget doesn't work out. The question, then, is why didn't oxygen continue to accumulate after the initial rise during the Great Oxygenation event? Cyanobacteria were still photosynthesizing. They were still producing oxygen. So where was it going? The answer lies in the balance between oxygen sources and oxygen sinks. Sources are processes that add oxygen to the atmosphere. Photosynthesis is the main one. Sinks are processes that remove oxygen. And during the boring billion, the sinks were powerful enough to match the sources almost perfectly. One major sink was the oxidation of continental surfaces. When oxygen first began to accumulate in the atmosphere, it encountered vast quantities of unoxidized rock. Iron-bearing minerals on land, particularly pyrite, reacted readily with oxygen. These reactions consumed enormous amounts of the gas, preventing it from building up in the atmosphere. Even after the initial wave of oxidation during the GOE, this process continued. Tectonic activity, though subdued during the boring billion, still brought fresh unoxidized rock to the surface over time. Erosion exposed new rock faces, and each time new rock was exposed, atmospheric oxygen reacted with it, keeping oxygen levels low. Another sink involved the burial of organic carbon. When photosynthetic organisms die, their remains can be buried in sediments. If they're buried before they decompose, the carbon in their bodies is effectively removed from the carbon cycle. This burial process also affects oxygen levels, because decomposition consumes oxygen. Less decomposition means less oxygen consumption, which should lead to oxygen accumulation. But during the boring billion, organic carbon burial rates appear to have been low. The nutrient scarcity we discussed earlier meant that marine productivity was limited. Fewer organisms meant less organic matter, and less organic matter meant less burial. The result was that this potential oxygen source never fully materialized. The oceans themselves also acted as an oxygen sink. The deep ocean, as we'll explore in detail shortly, was anoxic throughout most of the boring billion. Oxygen produced in shallow surface waters didn't penetrate into the depths. Instead, it was consumed by chemical reactions and microbial respiration before it could mix downward. The deep ocean remained a vast reservoir of oxygen debt. The net effect of all these processes was a plateau. Oxygen production and oxygen consumption balanced each other. The atmosphere held steady at low levels, and this plateau persisted for 1 billion years. There were probably variations, periods when oxygen rose slightly and periods when it fell, but the overall trend was flat. This oxygen ceiling had direct biological consequences. Organisms that depended on oxygen, including the early eukaryotes that were beginning to evolve during this time, were restricted to environments where oxygen was available. That meant shallow waters near the ocean surface, where photosynthesis occurred. The deeper you went, the less oxygen you found. Below a certain depth, there was none at all. This created a layered ocean, a thin, oxygenated surface layer where complex life could potentially exist, and a thick, anoxic deep layer where only anaerobic microbes could survive. Most of the ocean's volume was off-limits to oxygen-dependent organisms. They were confined to a narrow band of habitable water, hugging the continental shells where light penetrated and photosynthesis could occur. Even in that narrow band, oxygen levels were marginal, not absent, but low enough that metabolic rates were constrained. Organisms couldn't grow as large or as complex as they would in a high-oxygen world. The energy available to them was limited by the amount of oxygen they could access, and that amount remained stubbornly low throughout the boring billion. The oxygen ceiling, in other words, was both a chemical constraint and a spatial one. It limited where life could exist and what forms it could take. Complex organisms require oxygen, but during the boring billion, oxygen was scarce, and the ocean environments where it was available were small. Life was squeezed into a thin layer of habitable water, unable to expand either upward into a low-oxygen atmosphere or downward into anoxic depths. And so, evolution proceeded under these constraints, slowly, carefully, building the cellular machinery that would eventually allow organisms to exploit oxygen more efficiently. But for the moment, for hundreds of millions of years, that machinery was still being developed, and life remained small, simple, and confined to the narrow zones where oxygen could be found. If you could dive beneath the surface of a boring billion ocean, the journey downward would be a journey into a fundamentally different world. The sunlit surface waters, where light penetrates and photosynthesis occurs, would give way within a few dozen meters to a realm of permanent darkness. And in that darkness, the chemistry of the water would change in ways that made it hostile to nearly all forms of complex life. The ocean during this time was stratified, not just by temperature or salinity, but by chemistry. The upper layer, the photic zone where sunlight reached, contained some oxygen. Not much by modern standards, but enough to support photosynthetic cyanobacteria and perhaps some early eukaryotic organisms. This layer was thin, probably no more than 100 meters deep in most places. Below that, oxygen disappeared entirely. The water became anoxic. This wasn't unusual. Even in modern oceans, deep waters can be depleted of oxygen in certain regions where circulation is poor. But during the boring billion, anoxia wasn't confined to isolated pockets. It was the default state of the deep ocean almost everywhere across the planet. Anoxic water alone would have been challenging for complex life, but manageable. Some organisms can survive in low oxygen or even zero oxygen environments by switching to anaerobic metabolism. They slow down their activity, reduce their energy consumption, and wait for conditions to improve. But the deep waters of the boring billion weren't just anoxic. They were actively toxic. The toxicity came from sulfur, specifically from hydrogen sulfide, a compound produced by bacteria that metabolize sulfate in the absence of oxygen. Hydrogen sulfide is poisonous to most eukaryotic organisms. It interferes with cellular respiration by binding to the enzymes that cells use to process oxygen. Even small concentrations can be lethal. And in the deep oceans of the boring billion, hydrogen sulfide wasn't present in small concentrations. It was abundant. This state, where water is both anoxic and sulfidic, is called euxinia. The term comes from the ancient name for the Black Sea, which today has euxinic deep waters. But during the boring billion, euxinia wasn't limited to a single sea. It characterized the global deep ocean. Researchers refer to this planetary state as the Canfield Ocean, after the scientist who first proposed the model. The chemistry that created the Canfield Ocean was set in motion by the Great Oxygenation Event. When atmospheric oxygen first appeared, it reacted with sulfur-bearing minerals on land, particularly iron pyrite. These reactions released sulfate into rivers and eventually into the ocean. Sulfate concentrations in seawater increased dramatically following the GOE and remained high throughout the Proterozoic. In the oxygenated surface waters, sulfate was relatively inert, but when it sank into the anoxic depths, Microbes went to work on it. Sulfate-reducing bacteria use sulfate as an electron, acceptor in their metabolism, much like aerobic organisms use oxygen. The byproduct of this process is hydrogen sulfide, and because the deep ocean was anoxic, there was no oxygen available to oxidize the hydrogen sulfide back into sulfate. It accumulated. The result was a deep ocean that smelled of rotten eggs, the characteristic odor of hydrogen sulfide. The water would have had an unusual appearance as well. Modern researchers studying ancient ocean chemistry suggest that the euxinic waters of the boring billion may have appeared black in the deepest basins, where hydrogen sulfide concentrations were highest, transitioning to a milky turquoise color in shallower anoxic zones where sulfur compounds were present in different forms. This was an ocean that looked nothing like the blue or green waters we associate with healthy marine environments today. It was chemically alien, a vast body of water where the dominant chemistry was reducing rather than oxidizing, where sulfur cycling dominated carbon cycling, and where the conditions that support complex life simply did not exist. The transition between the oxygenated surface and the sulfidic depths would have been sharp, a boundary layer, perhaps only a few meters thick, where oxygen from above met hydrogen sulfide from below. In this layer, chemical reactions would have occurred constantly. Sulfur-oxidizing bacteria, which use oxygen to metabolize hydrogen sulfide, would have clustered along this boundary, taking advantage of the chemical gradient. But this boundary was also a barrier. Organisms adapted to the oxygenated surface couldn't penetrate into the sulfidic depths, and organisms adapted to the anoxic depths couldn't rise into the oxygenated surface. Each zone had its own community, its own ecology, and very little exchange occurred between them. The euxinic deep ocean represented a massive loss of habitable space. In a modern ocean, the deep sea is not particularly hospitable, but it is at least accessible to certain organisms. Deep-sea fish, invertebrates, and microbes thrive in the cold, dark waters below the sunlit surface. But during the boring billion, the deep ocean was functionally dead to any organism that depended on oxygen or that couldn't tolerate hydrogen sulfide. This meant that life was compressed into the uppermost layer of the ocean. All the biological activity, all the photosynthesis, all the early eukaryotic evolution, everything that was happening biologically, was confined to a thin skin of water at the top of a vast, toxic sea. The ocean had volume, enormous volume, but almost none of it was usable, and this condition persisted. Not for millions of years, but for hundreds of millions. The euxinic state became the normal state. Generations upon generations of organisms lived and died in the shallow waters, never knowing that the darkness below was anything other than poison. The deep ocean remained a chemical wasteland, cut off from the surface, cycling its sulfur, compounds in an endless, toxic loop. The descent into darkness, then, was not just a journey into the absence of light. It was a journey into a fundamentally inhospitable environment, a place where the basic chemistry of life, the chemistry that depends on oxygen and avoids sulfur toxicity, simply couldn't function. And for 1 billion years, that place comprised the vast majority of Earth's ocean. The concept of the Canfield Ocean represents one of the most significant shifts in how scientists understand the Proterozoic world. For decades, researchers assumed that the ancient oceans were simply low in oxygen, but otherwise similar to modern seas. The Canfield Ocean model, proposed in the late 1990s, revealed something far stranger. The deep ocean wasn't just oxygen-poor, it was sulfur-rich, and that difference mattered enormously. Donald Canfield, the geochemist for whom the model is named, arrived at this conclusion by studying the chemistry of ancient rocks. Specifically, he looked at the ratios of different sulfur isotopes preserved in sediments from the Proterozoic. These isotopes act as a chemical fingerprint, revealing the processes that were active when the rocks formed. What Canfield found was evidence of widespread sulfate reduction. The isotope ratios indicated that sulfate-reducing bacteria were incredibly active in the Proterozoic oceans, far more active than in the modern ocean. This suggested that the deep waters were not only anoxic, but also rich in sulfate, providing abundant fuel for sulfate-reducing microbes. And where there's active sulfate reduction, there's hydrogen sulfide production. The implications were profound. If the deep ocean was sulfidic, then it wasn't just an unproductive environment. It was actively harmful. Hydrogen sulfide is toxic to eukaryotes. It poisons the enzymes that allow cells to use oxygen. Even organisms that don't depend on oxygen are often negatively affected by hydrogen sulfide. Only specialized microbes, those with specific adaptations for handling sulfur compounds, can thrive in such conditions. This meant that the habitable volume of the ocean was drastically reduced. Not just reduced, but compressed into the shallowest possible zones. Life couldn't exist in the sulfidic depths. It couldn't exist in the anoxic mid-depths. It could only exist in the thin surface layer, where oxygen was present and hydrogen sulfide hadn't penetrated. The Canfield Ocean also had consequences for nutrient availability. Iron, an essential element for many biological processes, is highly reactive with sulfur. In water rich with hydrogen sulfide, dissolved iron immediately forms iron sulfide minerals and precipitates out of the water column. It sinks to the seafloor and is buried in sediments, effectively removed from circulation. This explains one of the puzzles of the boring billion. The geological record from this period lacks banded iron formations, the layered deposits of iron-rich minerals that are common in older Proterozoic rocks. For a long time, this absence was interpreted as evidence that the oceans had been depleted of iron. But the Canfield Ocean model offers a different explanation. The iron was still there, being delivered to the ocean by rivers, but it was being stripped out immediately by reaction with hydrogen sulfide in the deep water. The same process affected other metals. Molybdenum, zinc, copper, selenium, all bioessential trace elements, form insoluble compounds when they encounter hydrogen sulfide. These compounds precipitate rapidly and settle into the sediments. The euxinic deep ocean, in other words, acted as a chemical trap. It removed from circulation the very elements that life needed to build complex proteins and enzymes. This created a vicious cycle. The anoxic conditions in the deep ocean promoted sulfate reduction, which produced hydrogen sulfide. The hydrogen sulfide stripped metals from the water. The loss of metals limited biological productivity in the surface ocean. Lower productivity meant less organic matter sinking into the deep ocean, which meant less oxygen demand, which helped maintain the anoxic state. The system was self-reinforcing. Breaking out of this cycle required something to change at a fundamental level. Either oxygen levels needed to rise high enough to penetrate the deep ocean and oxidize the hydrogen sulfide, or something needed to disrupt the sulfur cycle itself. But during the boring billion, neither of these things happened. The system remained locked in its euxinic state. The biological communities that did exist in this ocean were dominated by organisms adapted to these conditions. In the surface waters, cyanobacteria performed oxygenic photosynthesis, producing oxygen that kept the upper layer habitable. But below them, in the transition zones and anoxic waters, different bacteria thrived. Anoxygenic photosynthetic bacteria, particularly purple sulfur bacteria, were likely abundant. These organisms use hydrogen sulfide instead of water as the electron donor for photosynthesis. They don't produce oxygen. Instead, they produce elemental sulfur or sulfate as a byproduct. These bacteria can thrive in low light conditions, living just below the zone where oxygen is present, feeding on the upwelling hydrogen sulfide from the depths. The presence of these purple bacteria would have given the ocean a distinctive colour. In zones where they were abundant, the water would have appeared purple or reddish, coloured by the photosynthetic pigments in the bacterial cells. Combined with the black and turquoise hues of the deeper sulfidic waters, the boring billion ocean would have been a patchwork of strange alien colours. This was an ocean fundamentally shaped by microbial metabolism. The chemistry of the water, the availability of nutrients, the colour of the sea, all were determined by the metabolic activities of bacteria. Eukaryotes were present, but they were marginal, confined to narrow niches where conditions were just barely tolerable. The ocean belonged to the prokaryotes, and it would remain their domain for hundreds of millions of years. The Canfield Ocean persisted throughout most of the boring billion, a stable planetary state that resisted change. The chemistry was self-sustaining, the biology adapted to it, and the physical structure of the ocean, stratified with a thin, oxygenated surface over a thick sulfidic deep, remained essentially unchanged. The Canfield Ocean, then, was not just a feature of the boring billion. It was one of the defining constraints, a chemical cage that limited where life could exist and what forms it could take. A planetary-scale phenomenon that shaped evolution for 1 billion years. Imagine standing on a shore during the Boring Billion, looking out over the ocean. The water before you would not shimmer blue or green as modern seas do. The colour would be different. Strange. In some places, perhaps a milky turquoise, the result of fine sulphur particles suspended in the water. In others, particularly over the deeper basins, a dark, almost black hue where light penetrated only a short distance before being absorbed by the chemistry of the water itself. The appearance of the ocean is determined by what's dissolved or suspended in it, and by what happens to light as it passes through. In modern oceans, the blue colour comes from the way water molecules scatter sunlight, preferentially scattering blue wavelengths. The green tints come from phytoplankton, microscopic photosynthetic organisms that contain chlorophyll. Clear tropical waters appear bright blue. Productive temperate waters appear greenish. But all of these colours depend on relatively clean, oxygenated water. The Boring Billion Ocean was neither clean nor fully oxygenated below the surface. In the shallow coastal zones where oxygen was present and cyanobacteria thrived, the water might have appeared somewhat normal, tinged green by photosynthetic microbes. But move offshore into deeper water, and the colour would shift. The milky turquoise appearance would have come from colloidal sulphur. When hydrogen sulfide from the deep ocean mixed with trace amounts of oxygen in the water column, chemical reactions occurred that produced elemental sulfur in the form of extremely fine particles. These particles, too small to settle quickly, remained suspended in the water, scattering light in unusual ways. The result was a cloudy, pale blue-green colour, unlike anything seen in modern oceans. Deeper still, where no oxygen penetrated and hydrogen sulfide concentrations were high, the water would have absorbed light more completely. Hydrogen sulfide itself is colourless, but the chemical environment it creates, rich in dissolved metals, organic compounds, and other sulfur species, affects how light behaves. The water would have appeared dark, even in relatively shallow depths. Visibility would have been poor. Looking down from the surface, you would see blackness. This darkness wasn't just a visual phenomenon, it had biological consequences. Photosynthesis requires light. Most photosynthetic organisms need to be in the upper 50 to 100 metres of the water column, where sunlight is sufficient. But in the Boring Billion Ocean, the light-penetrating depth may have been even shallower due to the particulate matter and dissolved compounds in the water. The productive zone, where photosynthesis could occur, was compressed. The texture of the water would have been different as well. Modern seawater has a certain feel, a smoothness that comes from its relatively simple chemical composition, mostly water with dissolved salts. But the Boring Billion Ocean carried a higher load of dissolved compounds. Sulfates, sulfides, metal ions, organic molecules. The water would have felt slightly heavier, slightly more viscous, though the difference would have been subtle. The smell, however, would not have been subtle at all. Hydrogen sulfide has a powerful odour, the smell of rotten eggs. In areas where euxinic water was close to the surface or where upwelling brought deep water into contact with the atmosphere, the smell would have been overwhelming. Coastal zones near deep basins might have been surrounded by this odour constantly, a reminder of the toxic chemistry lurking below. In some regions, particularly in shallow embayments or lagoons where circulation was restricted, the euxinic conditions might have extended all the way to the surface. These would have been dead zones in the truest sense. Water that was black, foul-smelling and completely devoid of oxygen. Nothing larger than sulfate-reducing bacteria could survive there. The water would have been still, undisturbed by swimming animals or drifting organisms, its surface perhaps covered with a thin sheen of sulfur compounds. But in other areas, particularly along the continental shelves where rivers delivered oxygen-rich water and where wave action mixed the surface, the water would have been more habitable. Here, the ocean might have appeared relatively normal, at least at first glance, green-tinged from photosynthetic life, clear enough to see a metre or two down. But even in these favourable zones, diving below the surface would quickly reveal the transition to anoxic and then euxinic conditions. The boundaries between these different water types would have been sharp. A chemocline, the chemical equivalent of a thermocline, marked the transition from oxygenated surface water to sulfidic deep water. Crossing this boundary would have meant entering a fundamentally different world. Above it, oxygen and life. Below it, hydrogen sulfide and microbial communities adapted to darkness. These boundaries might have been visible. Where oxygenated water mixed with sulfidic water, chemical reactions would produce clouds of precipitating minerals and elemental sulfur. These clouds would have appeared as hazy layers in the water column, marking the zones where chemistry changed. Swimming or diving through such a layer would have been like passing through a curtain of fine particles. The seasonal variation we see in modern oceans, blooms of phytoplankton in spring, clear water in winter, would have been muted during the boring billion. The nutrient limitation and climate stability meant that biological activity remained relatively constant throughout the year. There were no great pulses of productivity, no seasonal die-offs, just a steady, low level of microbial activity, year after year, century after century. The boring billion ocean, then, was not just chemically different from modern seas. It looked different, it smelled different, it felt different. Standing on those ancient shores, you would have known immediately that you were looking at an alien world. Water that was the wrong colour, air that carried the wrong scent, a sea that appeared dead in ways that modern oceans, even at their most depleted, never do. And yet, within that strange water, life persisted. Microbial communities adapted to conditions that would be instantly lethal to most modern organisms. Photosynthetic bacteria clustered in the thin surface layer. Sulfate reducers thrived in the dark depths. And somewhere in between, the first complex cells were beginning their slow, patient evolution towards something more. In the zones where oxygenated surface water transitioned to sulfidic depth, a particular group of organisms thrived. These were the purple sulfur bacteria, photosynthetic microbes that represent one of the most important, yet often overlooked players in the ecology of the boring billion ocean. They were adapted to conditions that most organisms would find intolerable, and they played a central role in maintaining the very chemical conditions that defined this era. Purple sulfur bacteria are anoxygenic photosynthetic organisms. That phrase requires some unpacking. Photosynthesis, in its most familiar form, uses water as an electron donor. Cyanobacteria and plants split water molecules, releasing oxygen as a byproduct while capturing the electrons for use in building organic compounds. This is oxygenic photosynthesis, and it's responsible for essentially all the oxygen in Earth's atmosphere. But there are other forms of photosynthesis. An oxygenic photosynthesis uses different electron donors. Purple sulfur bacteria use hydrogen sulfide. They take H2S, strip away electrons, and use those electrons to power their metabolism. The sulfur is left behind, either as elemental sulfur that accumulates inside or outside the cell, or as sulfate that's released into the water. Critically, no oxygen is produced in this process. This metabolic strategy allowed purple sulfur bacteria to occupy a niche that oxygenic photosynthesizers couldn't. They could live in the transition zone between oxygenated and sulfidic water, where hydrogen sulfide was present but light still penetrated. They could even survive in fully anoxic water, as long as some light reached them. This made them ideally suited for the boring billion ocean, where the deep water was rich in hydrogen sulfide and the surface water contained oxygen, with a transition zone between. The purple colour of these bacteria comes from their photosynthetic pigments. Unlike the green chlorophyll used by cyanobacteria and plants, purple sulfur bacteria use bacteria chlorophylls that absorb light in different wavelengths, particularly in the infrared and red parts of the spectrum. When present in high concentrations, these pigments give the water a distinctive purple or reddish tint. During the boring billion, purple sulfur bacteria were likely abundant in many marine environments. Anywhere that hydrogen sulfide-rich water came close to the surface, they would have formed dense blooms. Coastal upwelling zones, where deep water rose toward the surface, would have been particularly favourable. The purple bacteria would have thrived along the boundary between the oxygenated surface and the sulfidic depths, forming a layer of coloured water that marked the chemocline. Their presence had consequences beyond just colouring the water. By consuming hydrogen sulfide, purple sulfur bacteria acted as a buffer between the toxic deep ocean and the habitable surface. They processed the upwelling sulfide, converting it to less toxic forms before it could penetrate into the oxygenated zone. In doing so, they helped maintain the stratification of the ocean, keeping the surface waters relatively habitable while the depths remained sulfidic, but they also perpetuated the euxinic state. Unlike cyanobacteria, purple sulfur bacteria don't produce oxygen. They consume light and hydrogen sulfide, but they don't change the overall oxygen budget of the ocean. In fact, by thriving in the transition zone, they may have prevented oxygen from penetrating deeper. Any oxygen that diffused downward would be consumed by sulfur-oxidizing bacteria living alongside the purple bacteria before it could reach the deep sulfidic waters. This created a stable ecosystem structure. Cyanobacteria in the surface produced oxygen. Purple sulfur bacteria in the transition zone consumed hydrogen sulfide. Sulfate-reducing bacteria in the depths produced hydrogen sulfide. And the cycle continued, self-sustaining, for hundreds of millions of years. Each group of microbes was performing its metabolic function, and together they maintained the chemical stratification that defined the boring billion ocean. The purple bacteria also occupied a specific light environment. They can photosynthesize at lower light levels than cyanobacteria because their pigments are adapted to capture wavelengths that penetrate deeper into water. Red and infrared light travels farther through water than blue or green light. This allowed purple bacteria to live below the cyanobacteria in dimmer conditions where oxygenic photosynthesis couldn't occur. In some reconstructions of the boring billion ocean, researchers envision a layered photosynthetic system. Cyanobacteria at the top in the bright oxygenated water, producing oxygen and organic matter. Purple sulfur bacteria below them in the dim transition zone, using the hydrogen sulfide that diffused upward from the depths. And below that, darkness and euxinia. Each layer had its own ecology, its own chemical signature, and very little mixing between them. The dominance of purple sulfur bacteria during this time had another effect. It limited the production of oxygen. In a modern ocean, photosynthetic productivity is mostly oxygenic. Phytoplankton produce oxygen, which accumulates in the atmosphere. But during the boring billion, a significant fraction of photosynthesis may have been anoxygenic. The purple bacteria were productive, but they didn't contribute to atmospheric oxygen. This helped maintain the low oxygen levels that characterize stage 3 of Earth's oxygen history. There's evidence for this purple bacteria dominance in the geological record. Certain biomarker molecules, chemical fossils of ancient organisms, appear in sediments from the Proterozoic. Some of these biomarkers are specific to anoxygenic photosynthetic bacteria. Their presence suggests that these organisms were abundant and widespread, supporting the idea that the boring billion ocean had extensive zones, where anoxygenic photosynthesis dominated. The purple dominion, then, was both a biological and a chemical phenomenon. These bacteria thrived in conditions that other organisms couldn't tolerate. They helped maintain the euxinic state of the deep ocean by preventing oxygen penetration, and they represented a significant fraction of the ocean's primary productivity, yet contributed nothing to atmospheric oxygen. They were, in a sense, the ecological face of the boring billion, organisms perfectly adapted to a stable, constrained world, with no incentive to change and no ability to escape the system they help maintain. Life requires more than just carbon, hydrogen, oxygen, and nitrogen. These 4 elements form the backbone of biological molecules, but they're not sufficient alone. Organisms also need phosphorus for DNA and energy transfer. They need iron for electron transport. They need trace metals like molybdenum, zinc, copper, and selenium to build the enzymes that catalyze essential biochemical reactions. Without these elements, even the simplest cells cannot function. During the boring billion, many of these essential elements were vanishing from the ocean, not disappearing entirely, but being removed from circulation and locked away in sediments at rates faster than they could be replenished. The euxinic chemistry of the deep ocean was actively stripping the water of the very nutrients that life needed most. The mechanism was straightforward. Trace metals dissolve in water as ions, electrically charged atoms that remain suspended in solution. In oxygenated or mildly reducing conditions, many metal ions are relatively stable in this dissolved form. They can be transported by ocean currents, taken up by organisms, and cycled through the marine ecosystem. but in the presence of hydrogen sulfide, the chemistry changes. Most transition metals react rapidly with sulfide to form metal sulfide minerals. These minerals are highly insoluble. They precipitate out of solution almost immediately, forming tiny particles that sink to the seafloor and become buried in sediments. Once buried, they're effectively removed from the biosphere, locked away in rock. Iron was particularly affected. Iron sulfide, commonly known as pyrite when it forms the mineral FeS2, precipitates readily in sulfidic water. Any dissolved iron that entered the deep ocean, whether from hydrothermal vents, sediment dissolution, or mixing from surface waters, would encounter hydrogen sulfide and immediately form iron sulfide minerals. The result was that the deep ocean became severely depleted in dissolved iron. This explains the absence of banded iron formations during the boring billion. These distinctive layered rocks, which are common in older Proterozoic sediments, form when dissolved iron in seawater oxidizes and precipitates. But if iron is being stripped from the water column by reaction with hydrogen sulfide, it never gets the chance to form these deposits. The iron is still entering the ocean from rivers and hydrothermal sources, but it's being removed via a different pathway. Molybdenum faced a similar fate. This element is critical for nitrogen fixation, the process by which certain bacteria convert atmospheric nitrogen into forms that other organisms can use. Molybdenum is also a key component of enzymes involved in sulfur metabolism and other essential processes. But like iron, molybdenum forms insoluble sulfide minerals in euxinic water. The deep ocean acted as a massive sink, removing molybdenum from circulation. Analyses of ancient sediments from the early part of the boring billion, particularly between 1.8 and 1.4 billion years ago, show remarkably low concentrations of molybdenum and other trace metals. The ocean was depleted, not just in the deep basins where euxinic conditions prevailed, but even in some shallow water environments, because the deep ocean was such an effective sink that it drew down the global inventory of these elements. Zinc, copper, and selenium followed similar patterns. All of these elements form sulfide minerals. All were scarce during the boring billion, and all are essential for eukaryotic metabolism. Zinc, for example, is a cofactor in hundreds of enzymes. It's involved in DNA replication, gene expression, and immune function in modern organisms. Without adequate zinc, cells struggle to perform basic functions. The scarcity of these elements created a metabolic bottleneck. Even if an organism had access to sunlight, water, and carbon dioxide for photosynthesis, it still needed trace metals to build the enzymes that make photosynthesis work. Without molybdenum, nitrogen fixation becomes impossible. Without iron, electron transport chains can't function. Without zinc, DNA polymerase can't replicate genetic material efficiently. This was a fundamentally different kind of limitation than a simple lack of food or energy. It was a limitation at the molecular level. Organisms couldn't build the cellular machinery they needed because the building blocks, the metal cofactors, weren't available. Evolution could produce new genetic innovations, but those innovations couldn't be expressed if the necessary metals weren't present in the environment. The situation was compounded by the slow weathering rates we discussed earlier. Continental erosion was delivering trace metals to the ocean at reduced rates because the continents were flat and tectonic activity was low. At the same time, the euxinic deep ocean was removing metals at high rates. The input was low, the removal was high. The result was severe depletion. Phosphorus, while not a metal, faced its own scarcity issues. Some researchers have proposed that the biological boom of the Paleo-Proterozoic, which produced the oxygen that initiated the Great Oxygenation event, consumed much of the ocean's phosphorus. Photosynthetic bacteria incorporated phosphorus into their cells, and when they died, that phosphorus sank into sediments and was buried. This created a phosphorus debt that took hundreds of millions of years to repay. The slow weathering of the boring billion wasn't delivering enough phosphorus to make up for this deficit. Phosphate minerals weather slowly under the best conditions, and with low topography and subdued erosion rates, phosphorus input to the ocean was minimal. Life was running on a phosphorus budget that allowed survival but not expansion. The combined effect of these nutrient limitations was a marine biosphere operating far below its potential productivity. In modern oceans, certain regions are limited by iron availability, others by nitrogen, others by phosphorus. These limitations can be relieved by upwelling or river input that delivers the missing nutrient. But during the boring billion, the ocean was simultaneously limited by multiple nutrients across most environments. This created what ecologists call co-limitation. It wasn't just one nutrient constraining growth, but several at once. And co-limitation is particularly restrictive because you can't solve the problem by adding more of one nutrient. You need to increase the availability of all the limiting nutrients simultaneously. The vanishing elements, then, represented one of the most fundamental constraints on evolution during the boring billion. The ocean wasn't just nutrient poor, it was actively hostile to nutrient retention. The chemistry of the deep water was stripping essential elements from circulation, and the slow geological processes that might have replenished those elements weren't operating fast enough to make a difference. Life, confined to the surface waters and starved of essential nutrients, had no choice but to become extraordinarily efficient. Organisms evolved mechanisms to scavenge every available atom of iron, zinc, and molybdenum. They developed enzymes that could function with lower metal concentrations. They streamlined their metabolic pathways to minimize waste. But these were survival strategies, not pathways to complexity. For 1 billion years, the elements that life needed most were vanishing into the sulfidic depths, and life, unable to retrieve them, had to make do with what little remained. In an ocean where the depths were toxic and nutrients were scarce, life clustered in the only environments that offered both safety and sustenance. These were the shallow coastal zones, the narrow bands of water that lay over the continental shelves. Here, sunlight penetrated to the bottom. Rivers delivered fresh water and dissolved minerals. Wave action mixed oxygen into the water, and crucially, the water was shallow enough that the euxinic conditions of the deep ocean couldn't establish themselves. Continental shelves are the submerged edges of continents, the gently sloping underwater plains that extend from the shoreline to the point where the seafloor drops away into the deep ocean basins. In the modern ocean, these shelves are some of the most productive environments. They host coral reefs, kelp forests, and rich fisheries. During the boring billion, they served a similar role, though the organisms that inhabited them were far simpler. The key advantage of shelf environments was oxygenation. In deep water, oxygen produced at the surface is consumed by microbial respiration and chemical reactions before it can mix downward. But in shallow water, the entire water column can remain oxygenated. Wave action and wind mixing ensure that oxygen is distributed throughout, from the surface to the seafloor. This creates a refuge for organisms that depend on oxygen. Shelf environments also receive nutrient input from rivers. When rain falls on continents and flows downhill, it carries dissolved minerals from the weathering of rock. These rivers empty into coastal waters, delivering nutrients that would otherwise remain locked in continental interiors. During the boring billion, when continental weathering was slow, this riverine input was modest, but it was still more than what was available in the open ocean far from land. The shallow depth meant that photosynthetic organisms could exploit a larger fraction of the water column. In the open ocean, photosynthesis is confined to the upper 100 meters or so. Below that, insufficient light penetrates for photosynthesis to occur. But on a shallow shelf, if the water is only 20 or 30 meters deep, then photosynthetic microbes and mats can cover the seafloor itself, taking advantage of light that reaches all the way to the bottom. These seafloor communities would have been dominated by microbial mats. These are layered structures formed by different types of bacteria living in close association. At the top, cyanobacteria would have performed oxygenic photosynthesis, producing oxygen and organic matter. Below them, other bacteria would have consumed that organic matter, creating chemical gradients within the mat. The mats would have appeared as wrinkled, textured surfaces covering the seafloor, coloured green or brown by photosynthetic pigments. Over time, these microbial mats could build upward, forming structures called stromatolites. These are layered, rock-like formations created by the trapping and binding of sediment particles by the sticky surfaces of the bacterial mat. As the mat grows upward, it captures sediment, then grows through the sediment layer, then captures more sediment, creating a laminated structure that can persist in the rock record. Stromatolites reached their maximum diversity and abundance during the Mesoproterozoic era, the core of the boring billion. The fossil record shows that these structures were widespread in shallow marine environments across the globe. They occupied the coastal zones of the supercontinents, forming extensive reef-like structures in some areas. Their abundance suggests that microbial mat communities were thriving in these shallow refuges, even as the rest of the ocean remained relatively barren. But even these productive shallow zones were constrained. The water was oxygenated, but oxygen levels were still low by modern standards. Nutrients were more available than in the open ocean, but still scarce in absolute terms. And the threat of the deep ocean was always present. If circulation patterns shifted and brought sulfidic water up onto the shelf, it could poison the entire environment. There's evidence that this happened periodically. Some sedimentary rocks from the boring billion show laminations that indicate periodic anoxia, moments when oxygen depleted, or even euxinic water, intruded onto the continental shelf. These events would have been catastrophic for any oxygen-dependent organisms in the area. The microbial communities would have been disrupted. Stromatolites might have stopped growing temporarily. Only the most tolerant organisms would have survived. But after these events passed and oxygenated conditions returned, life would have recolonized. The shallow refugees were resilient. As long as some oxygenated water remained, photosynthetic communities could rebuild. The stromatolites would begin growing again. The microbial mats would re-establish themselves. Life persisted, even in the face of periodic stress. These shallow zones were also where the first eukaryotes likely made their home. Complex cells, with their higher metabolic demands and sensitivity to hydrogen sulfide, couldn't survive in the deep ocean. But in the oxygenated shallows, they had a chance. The water might not have been ideal, oxygen was low, nutrients were scarce, but it was habitable, and habitable was enough. The earliest fossil evidence of eukaryotes comes from rocks deposited in shallow marine environments. These fossils, often preserved as compressed organic films in fine-grained sediments, show the distinctive morphology of eukaryotic cells. They're larger than typical bacteria, with more complex internal structures. Their presence in shallow water deposits suggests that this is where eukaryotes first established themselves during the boring billion. The continental shelves then served as incubators. They provided the stable, moderately oxygenated environments where complex life could slowly develop. They were not abundant environments by modern standards. They didn't host the diversity or productivity that we associate with shallow seas today. But they were refuges, safe zones in an otherwise hostile ocean, and for 1 billion years they were where life held on, slowly refining the cellular machinery that would eventually allow it to break free from these constraints. The extent of these refuges was limited by the total length of continental coastline. More coastline meant more shallow shelf habitat. Less coastline meant fewer refuges, and as we'll see, changes in continental configuration during the boring billion had profound effects on the availability of these crucial shallow environments. The breakup and reassembly of supercontinents didn't just reorganize the geography of land, it reorganized the geography of life's refuges. If you could walk along a shallow seafloor during the boring billion, carefully making your way through water perhaps waist-deep or chest-deep, you would see structures rising from the sediment. Not plants, not animals, but layered mounds and columns of rock, built slowly over centuries by communities of bacteria. These were stromatolites, and they dominated the shallow marine environments of the Mesoproterozoic era in a way they never would again. Stromatolites are not living organisms in themselves. They're structures created by microbial communities. The bacteria that build them secrete sticky compounds that trap sediment particles. As the sediment accumulates, the bacteria grow upward through it, creating a new layer of living mat on top of the trapped sediment. Then they trap more sediment and grow upward again. Repeat this process thousands of times and you build a layered structure that can be meters tall and persist for millennia. The bacteria don't build stromatolites intentionally. They're not constructing anything in the way that termites build mounds or birds build nests. They're simply growing in response to light and nutrients, and the stromatolite is a byproduct of that growth combined with sediment trapping. But the result is one of the most distinctive structures in the fossil record. During the boring billion, stromatolites reached their peak diversity. Different environments produced different stromatolite forms. In calm, protected lagoons, they grew as broad, flat mats with subtle undulations. In areas with moderate wave action, they formed domical mounds. In zones with strong currents, they grew as elongated columns oriented in the direction of water flow. The morphology reflected the physical conditions of the environment. Some stromatolites grew individually, as isolated pillars rising from the seafloor. Others formed in clusters, creating reef-like structures where dozens or hundreds of columns grew close together. Still others formed as continuous sheets, coating large areas of the seafloor with layered, wrinkled surfaces. The variety was remarkable, and paleontologists have catalogued numerous distinct stromatolite forms from Mesoproterozoic rocks. The bacteria that built these structures were primarily cyanobacteria, though other types of microbes were also present in the mats. Cyanobacteria perform oxygenic photosynthesis, so they need sunlight. This restricted stromatolites to shallow water, where light could penetrate. The optimal depth varied depending on water clarity, but generally stromatolites formed in water less than 50 meters deep, with most in much shallower zones. The internal structure of stromatolites reveals the seasonal and environmental variations that occurred during their growth. Each layer represents a period of growth followed by sediment deposition. The thickness of layers varies, sometimes dramatically, suggesting that growth rates changed in response to environmental conditions. Thicker layers might indicate periods of rapid growth during favorable conditions. Thinner layers might represent times when growth slowed due to limited nutrients or light. Some stromatolites show evidence of periodic disturbance, layers that are disrupted or incomplete, suggesting that storms or other physical events damaged the microbial mat. But above these disturbed layers, you can see where growth resumed, with new layers forming on top of the damaged ones. The stromatolites were resilient, able to recover from physical damage as long as the underlying bacterial community survived. The microbial communities within stromatolites were vertically stratified. Different bacterial species occupied different depth levels within the mat, each adapted to specific chemical conditions. At the very top, in direct sunlight, cyanobacteria dominated. Just below, where light was dimmer, you might find purple sulfur bacteria using anoxygenic photosynthesis. Deeper still, in the anoxic interior of the mat, sulfate-reducing bacteria would be metabolizing sulfate and producing hydrogen sulfide. This hydrogen sulfide could then diffuse upward, providing fuel for the purple bacteria above. The mat, in other words, was a self-contained ecosystem, a miniature version of the layered ocean with oxygenated surface zones transitioning to sulfidic depths, but compressed into a structure only centimeters thick. Each layer of bacteria modified the chemical environment around it, creating conditions that allowed other bacterial types to thrive. The system was interconnected, with the waste products of one species serving as nutrients for another. Stromatolites played an important ecological role beyond just providing habitat for bacteria. They stabilized sediments, preventing them from being washed away by currents. They created topographic complexity on otherwise flat seafloors, providing surfaces for other microbes to colonize. And they produced oxygen, locally enriching the water immediately around them. But their abundance during the boring billion also tells us something about the state of the ecosystem. In modern oceans, stromatolites are rare. They're confined to a few extreme environments, like the hypersaline waters of Shark Bay in Australia, where grazing animals can't survive. The reason modern stromatolites are rare is that animals eat them. Grazing organisms consume the microbial mat, preventing it from accumulating enough to build a stromatolite structure. During the boring billion, there were no grazers. No animals existed yet. The microbial mats could grow undisturbed for as long as environmental conditions remained favorable. This allowed stromatolites to flourish. Their peak diversity during this time reflects not just favorable conditions for their growth, but the absence of predation pressure. They dominated because nothing could stop them. The decline of stromatolites in later geological periods, after the boring billion ended, coincides with the rise of animals capable of grazing on microbial mats. Once grazing evolved, stromatolites were largely restricted to marginal environments where grazers couldn't thrive. The boring billion, then, represents the last great age of stromatolites, a time when these structures covered vast areas of shallow seafloor without interference. Walking through those ancient shallow seas, you would have been surrounded by these gardens, columns and mounds rising around you, their surfaces slick with bacterial slime, coloured in shades of green and brown and sometimes purple. The water would have been clear in some areas, cloudy in others, depending on how much sediment was suspended. And everywhere, the slow work of bacterial growth, building structures that would persist in rock for billions of years, long after the bacteria themselves had died. The Stromatolite Gardens of the boring billion were monuments to patience. Each structure represented centuries or millennia of continuous bacterial activity. They grew slowly, imperceptibly, adding layers at rates measured in millimeters per year or slower. But over vast spans of time, they transformed the seafloor, creating landscapes unlike anything in the modern ocean. They were the visible face of a microbial world, proof that even in the absence of animals and plants, life could create beauty and complexity. Somewhere in the shallow coastal waters of the early boring billion, a different kind of cell appeared. It was larger than the bacteria that dominated the ocean. Its internal structure was more complex, with membrane-bound compartments separating different cellular functions. Most notably, it had a nucleus, a central chamber where genetic material was organized and protected. This was a eukaryotic cell, and its appearance marked one of the most significant transitions in the history of life. The origin of eukaryotes is one of the most studied questions in evolutionary biology, and also one of the most difficult to answer with certainty. The oldest fossils that are unambiguously eukaryotic appear in rocks from around 1.8 billion years ago, right at the beginning of the boring billion. But molecular clock analyses, which use genetic data to estimate when different lineages diverged, suggest that eukaryotes may have originated even earlier, perhaps by 2 billion years ago. What's clear is that by the start of the boring billion, eukaryotes existed. They were not abundant, they were not diverse, but they were present, living alongside the bacterial and archaeal cells that had dominated Earth for more than 1 billion years. And unlike those simpler cells, eukaryotes carried within them the potential for complexity. The defining feature of a eukaryotic cell is compartmentalization. Prokaryotes, bacteria and archaea, have relatively simple internal organization. Their genetic material floats freely in the cytoplasm. Their metabolic machinery is distributed throughout the cell. Eukaryotes, by contrast, divide their internal space into specialized compartments. The nucleus houses DNA. Mitochondria produce energy. In photosynthetic eukaryotes, chloroplasts perform photosynthesis. This compartmentalization allows for more sophisticated regulation of cellular processes. This complexity comes at a cost. Eukaryotic cells require more energy to maintain than prokaryotic cells. They're larger, which means they have more membrane surface area to maintain and more internal volume to manage. They have more complex genomes that require more resources to replicate, and they typically reproduce more slowly than bacteria, which can divide every 20 minutes under ideal conditions while eukaryotes might take hours or days. Given these costs, why did eukaryotes evolve at all? The answer lies in the advantages that complexity provides. Eukaryotes can regulate their internal chemistry with greater precision than prokaryotes. They can sequester toxic compounds in specific compartments. They can evolve specialized structures for specific functions. And crucially, they can become larger, which opens up new ecological niches. But during the boring billion, most of these advantages were muted. The ocean was nutrient poor, which limited how large cells could grow. Oxygen was scarce, which constrained the energy available for complex metabolism. And the dominance of microbial mat communities meant that niches for larger, more complex organisms were limited. Eukaryotes existed, but they remained marginal. The most important aspect of early eukaryotic evolution was the acquisition of mitochondria. Mitochondria are organelles that produce energy through aerobic respiration, but they weren't originally part of the eukaryotic cell. They were once free-living bacteria that were engulfed by an ancestral eukaryote and then became permanent residents, providing energy in exchange for shelter and nutrients. This process, called endosymbiosis, was transformative. Mitochondria allowed eukaryotes to extract far more energy from organic molecules than was possible through anaerobic metabolism. With this energy surplus, eukaryotic cells could afford to maintain complex internal structures. They could grow larger. They could support more sophisticated cellular machinery. Mitochondria, in a sense, were the power plants that made eukaryotic complexity possible. The timing of mitochondrial acquisition is debated. Some evidence suggests it happened very early, perhaps before the boring billion began. Other evidence suggests it occurred during the early part of this era. What's certain is that by the time eukaryotes begin to appear more prominently in the fossil record, they already have mitochondria. The two, eukaryotic complexity and mitochondrial power, appear to have been inseparable, but having mitochondria also created a dependency on oxygen. Mitochondria use oxygen to generate energy. Without it, they can't function efficiently. This meant that eukaryotes were tied to oxygenated environments even more strictly than most prokaryotes. They couldn't survive in the anoxic depths. They couldn't tolerate sulfidic water. They were restricted to the shallow, oxygenated refuges where oxygen, however limited, was available. During the boring billion, this restriction severely limited eukaryotic distribution. They couldn't colonize the deep ocean. They couldn't thrive in the transition zones where purple bacteria dominated. They were confined to the shallowest coastal waters, competing with cyanobacteria and other prokaryotes for the limited nutrients available there. The fossil record from the early boring billion shows simple eukaryotic cells. They appear as organic walled microfossils, preserved as compressed films in sedimentary rocks. Their morphology is distinctive, larger than bacteria, with smooth or ornamented cell walls, sometimes showing signs of internal structure. But they're not doing anything dramatically different from the prokaryotes around them. They're simply existing, persisting in an environment that barely supports them. This persistence, however, was crucial. While the environment wasn't favorable for eukaryotic diversification, it also wasn't eliminating them. They held on in their narrow niches, maintaining their complex cellular machinery, passing their mitochondria to daughter cells with each division. And within those cells, genetic evolution continued, slowly, invisibly, but continuously. The eukaryotic cell, during the boring billion, was a solution waiting for the right problem. It was a more sophisticated, more energetically expensive design than prokaryotic cells. In the stable, resource-poor ocean, there was no immediate advantage to this sophistication. Bacteria could survive just as well, if not better, with their simpler designs. But the eukaryotic cell carried within it the genetic toolkit for future complexity. Multicellularity would require eukaryotes. Sexual reproduction would require eukaryotes. The differentiation of specialized cell types, the foundation of plants, animals, and fungi, would require eukaryotes. None of these innovations could occur in prokaryotic cells, which lack the internal complexity and regulatory machinery to coordinate such developments. The appearance of eukaryotes during the boring billion, then, was the planting of a seed that wouldn't germinate for hundreds of millions of years. But it was planted. And it survived. In the shallow refuges among the Stromatolite Gardens, eukaryotic cells floated and divided and passed on their complex internal architecture to the next generation. They were rare. They were marginal. But they were there, carrying within them the potential for everything that would follow. Evolution, when viewed from the perspective of deep time, often appears to accelerate during periods of environmental upheaval. Mass extinctions, climate shifts, and ecological disruptions create opportunities for rapid adaptation and diversification. But the boring billion suggests a different pattern. Here, evolution advanced not through dramatic change, but through sustained pressure applied over unimaginable spans of time. The environmental constraints of this era, low oxygen, scarce nutrients, euxinic depths, tectonic stability, created a situation where survival itself required innovation. Organisms couldn't simply wait for better conditions. The conditions weren't going to improve for hundreds of millions of years. Instead, life had to develop strategies to extract every possible advantage from the resources available. For eukaryotes, this meant developing extraordinary efficiency. In a nutrient-poor environment, cells that could acquire and retain nutrients more effectively had a selective advantage. This drove the evolution of more sophisticated nutrient uptake mechanisms. Cells developed specialized membrane proteins that could scavenge trace metals from the water with high efficiency. They evolved internal storage systems that could hold onto nutrients when they were available, then ration them out slowly during lean times. The scarcity of trace metals was particularly challenging. These elements are required in tiny amounts, but they're absolutely essential. Without them, key enzymes can't function. Cells evolved regulatory systems that could sense metal availability and adjust metabolism accordingly. When molybdenum was scarce, some organisms evolved alternative enzymes that could perform similar functions using different metal cofactors. When iron was limited, cells developed mechanisms to recycle iron from old proteins rather than constantly requiring fresh input. This metabolic flexibility came at a cost. More complex regulation requires more genetic material and more protein machinery. It's energetically expensive to maintain these systems, but in an environment where the alternative was starvation, the investment was worthwhile. The cells that survived were those that could operate efficiently under extreme nutrient limitation. One of the most significant innovations that likely emerged or was refined during this period was sexual reproduction. Sexual reproduction is metabolically expensive and genetically complex. It requires cells to produce specialized evolution when viewed from the perspective of deep time, often appears to accelerate during periods of environmental upheaval. Mass extinctions, climate shifts, and ecological disruptions create opportunities for rapid adaptation and diversification. But the boring billion suggests a different pattern. Here, evolution advanced not through dramatic change, but through sustained pressure applied over unimaginable spans of time. The environmental constraints of this era, low oxygen, scarce nutrients, euxinic depths, tectonic stability, created a situation where survival itself required innovation. Organisms couldn't simply wait for better conditions. The conditions weren't going to improve for hundreds of millions of years. Instead, life had to develop strategies to extract every possible advantage from the resources available. For eukaryotes, this meant developing extraordinary efficiency. In a nutrient-poor environment, cells that could acquire and retain nutrients more effectively had a selective advantage. This drove the evolution of more sophisticated nutrient uptake mechanisms. Cells developed specialized membrane proteins that could scavenge trace metals from the water with high efficiency. They evolved internal storage systems that could hold onto nutrients when they were available, then ration them out slowly during lean times. The scarcity of trace metals was particularly challenging. These elements are required in tiny amounts, but they're absolutely essential. Without them, key enzymes can't function. Cells evolved regulatory systems that could sense metal availability and adjust metabolism accordingly. When molybdenum was scarce, some organisms evolved alternative enzymes that could perform similar functions using different metal cofactors. When iron was limited, cells developed mechanisms to recycle iron from old proteins rather than constantly requiring fresh input. This metabolic flexibility came at a cost. More complex regulation requires more genetic material and more protein machinery. It's energetically expensive to maintain these systems, but in an environment where the alternative was starvation, the investment was worthwhile. The cells that survived were those that could operate efficiently under extreme nutrient limitation. One of the most significant innovations that likely emerged or was refined during this period was sexual reproduction. Sexual reproduction is metabolically expensive and genetically complex. It requires cells to produce specialized reproductive cells, find mates, and combine genetic material in ways that create genetic diversity in offspring. For organisms living in stable environments where the same strategies work, generation after generation, sexual reproduction offers little advantage. Asexual reproduction, simply copying yourself, is faster and more efficient. But the boring billion, despite its apparent stability, was not truly stable at the organismal level. Local environments changed. Nutrient availability fluctuated. Oxygen concentrations varied with depth and season. In such conditions, genetic diversity becomes valuable. Offspring that are genetically different from their parents might be better suited to handle local environmental variations. Sexual reproduction creates this diversity. The evolution of sexual reproduction during the boring billion makes sense from this perspective. The environmental constraints were sustained and severe, but not uniform. There was variation in space and time, enough to favour genetic mixing and recombination, and once sexual reproduction evolved, it provided eukaryotes with a powerful tool for adaptation that prokaryotes lacked. Multicellularity represents another major innovation that likely originated during this time. Simple multicellularity, cells sticking together after division, is relatively easy to achieve and has evolved independently many times in different lineages. But true multicellularity, where cells differentiate into specialized types and coordinate their activities, is more complex. The advantage of multicellularity in a resource-poor environment is less obvious than in resource-rich ones. Large multicellular organisms need more nutrients than small single-celled ones. But multicellularity does offer some benefits even under constraint. A colony of cells can create chemical gradients within itself, allowing some cells to specialize in nutrient uptake, while others focus on other functions. This specialization can increase overall efficiency. Additionally, larger organisms are less vulnerable to being consumed by predators. While there were no animal predators during the boring billion, there were likely protist predators, larger eukaryotic cells that engulfed smaller ones. A multicellular cluster would be too large for these predators to consume, providing protection. The earliest evidence of multicellular eukaryotes appears in rocks from later in the boring billion, though the exact timing is uncertain due to the difficulty of fossilization and the ambiguity of around 1.46 billion years ago, something began to change. The supercontinent Columbia, which had persisted in stable configuration for hundreds of millions of years, started to break apart. The process was slow, even by geological standards. Rifts opened gradually. Continental fragments separated at rates measured in centimeters per year. But over tens of millions of years, those centimeters accumulated into thousands of kilometers. The configuration of Earth's surface was being remade. Continental breakup is driven by forces deep within the planet. Rising plumes of hot mantle material push upward against the base of the crust, creating tension. The continental lithosphere stretches, thins, and eventually ruptures. Magma wells up into the gap, forming new oceanic crust. The continental fragments drift apart, carried on the slowly moving tectonic plates. This process, called rifting, has occurred many times in Earth's history. But the lunar fragmentation event was particularly significant because of its timing and its consequences for life. The first signs of fragmentation would have been volcanic. Rifts are zones of intense igneous activity where magma reaches the surface and erupts. Long linear chains of volcanoes would have formed along the incipient rift zones, releasing lava and volcanic gases into the atmosphere. These eruptions would have been the most dramatic geological events in hundreds of millions of years, breaking the long volcanic quiescence that characterized the stable supercontinent phase. As the rifts widened, seawater would have flooded into the gaps. New ocean basins formed between the separating continental blocks. These young oceans were initially narrow, similar to the modern Red Sea, which is forming today as Africa and Arabia separate. Over time, as the continents continued to drift apart, these narrow seaways expanded into broad ocean basins. The fragmentation of Columbia transformed the planet's geography. A single large continent became several smaller ones, distributed across different latitudes. The total amount of land remained the same, but its configuration changed dramatically, and with that change came a fundamental shift in the relationship between land and sea. The most important consequence was the expansion of continental shelf habitat. Continental shelves are the shallow, submerged margins of continents. They're created by the gentle slope of continental crust as it transitions to oceanic crust. When a supercontinent is intact, the total length of continental margin, and thus the total extent of shelf habitat, is minimized. The land is concentrated in one place, with one continuous coastline. But when the supercontinent breaks apart, each fragment creates its own coastline. The total length of continental margin increases dramatically. Paleogeographic reconstructions suggest that the lunar fragmentation doubled the total length of stable continental shelf, expanding it to approximately 130,000 kilometers. This represents an enormous increase in the shallow marine habitat available for life. Shallow shelves are critical for marine ecosystems. They're where sunlight reaches the seafloor, enabling photosynthesis across the entire water column. They're where rivers deliver nutrients from continental weathering. They're where oxygen can penetrate throughout the water, creating refugees from the anoxic and euxinic conditions of the deep ocean. During the boring billion, when these refugees were the only habitable zones for complex life, doubling their extent was transformative. The timing of this expansion, around 1.46 billion years ago, aligns closely with the transition between the two phases of the slingshot hypothesis. The first phase, characterized by extreme nutrient scarcity and maximum constraint, ended around 1.4 billion years ago. The second phase, with its slightly improved conditions and beginning of eukaryotic diversification, began shortly after. The fragmentation event appears to have been the trigger for this transition. More shelf habitat meant more space for coastal communities. Stromatolites, which were already abundant, could expand their range. Microbial mats colonized the newly available seafloor. And critically, eukaryotes gained more territory. The shallow oxygenated zones where they could survive became more extensive, providing more opportunities for populations to establish and diversify. The fragmentation also affected ocean circulation. A single supercontinent creates a relatively simple circulation pattern, with ocean currents flowing around the continental mass. But when the continent breaks apart, currents can flow between the fragments, creating more complex circulation patterns. This can enhance the mixing of surface and deep waters in some regions, potentially bringing nutrients up from depth. However, this enhanced circulation was a double-edged benefit. While it could bring nutrients to the surface in some areas, it could also bring euxinic water from the deep ocean up onto the continental shelves. There's evidence in the rock record of periodic anoxic events during and after the fragmentation, moments when oxygen-depleted water invaded shelf environments. These events would have been stressful for oxygen-dependent life, but they were temporary. Once circulation patterns stabilized, the shelves returned to their role as oxygenated refugees. The breakup of Columbia didn't happen uniformly or all at once. Different parts of the supercontinent separated at different times. Some fragments remained connected by narrow land bridges even as other sections drifted apart. The process unfolded over perhaps 200 million years, from the initial rifting around 1.5 billion years ago to the final separation around 1.3 billion years ago. During this extended period of fragmentation, the planet existed in a transitional state, neither a unified supercontinent nor a collection of fully separated continents, but something in between. This transitional geography created a variety of marine environments, from narrow seaways with restricted circulation to broad shelves with good connection to the open ocean. This environmental diversity may have promoted biological diversity as well, providing a range of habitats where different adaptations could succeed. The volcanic activity associated with rifting also had atmospheric consequences. Volcanic eruptions release carbon dioxide, which acts as a greenhouse gas. The increase in volcanic outgassing during the fragmentation may have temporarily warmed the climate, offsetting some of the cooling caused by other processes. The climate remained stable overall, but there were likely minor fluctuations that created additional environmental variation. By 1.3 billion years ago, the fragmentation was largely complete. The former supercontinent Columbia existed as several distinct continental blocks, separated by ocean basins. The planet's geography had been fundamentally reorganized, and life, which had endured hundreds of millions of years of constraint in limited shallow refuges, now had access to twice as much habitable space. This expansion of habitat didn't immediately lead to an explosion of diversity. The constraints of low oxygen, limited nutrients, and euxinic deep water remained, but it provided room for the innovations that had been developing under pressure to spread. The slingshot, which had been pulled back by extreme scarcity, was beginning to release. The fragmentation was the first step in that release, creating the spatial conditions necessary for the next phase of evolution. The doubling of continental shelf length that resulted from the Nuna fragmentation created something precious during the boring billion. Space. Not just physical space, but ecological space. room for populations to expand, for communities to diversify, for evolution to experiment without immediately running into resource limitations. The expanded shallows represented the first significant increase in habitable marine environment in hundreds of millions of years. Understanding why this mattered requires understanding the concept of ecological carrying capacity. Every environment can support only a certain number of organisms, determined by the availability of resources: food, nutrients, space, light. All of these factors limit how many individuals can survive in a given area. When a population reaches the carrying capacity of its environment, growth stops, competition intensifies, organisms struggle for the limited resources available. During the early boring billion, marine life was pressed against the carrying capacity of the available shallow shelf habitat. The total extent of oxygenated, sunlit, relatively nutrient-rich water was limited. Every organism that could live in these environments was already there. There was no room for expansion, no opportunity for new forms to establish themselves without displacing existing ones. The fragmentation changed this equation. By doubling the shelf area, it doubled the carrying capacity. Suddenly there was room for populations to grow. Organisms could disperse into new territories without immediately encountering resource competition. And in these new territories, local conditions might be slightly different. Different water depths, different current patterns, different proximities to river mouths, creating opportunities for local adaptation. The expanded shallows were not uniform. Some shelf regions were broad and gently sloping, with extensive areas of very shallow water. Others were narrower, dropping off more steeply into the deep ocean. Some were adjacent to river deltas, receiving regular inputs of fresh water and nutrients. Others were far from major river systems, more nutrient-poor but also more stable in salinity and chemistry. The doubling of continental shelf length that resulted from the Nuna fragmentation created something precious during the boring billion: space. Not just physical space, but ecological space. Room for populations to expand, for communities to diversify, for evolution to experiment without immediately running into resource limitations. The expanded shallows represented the first significant increase in habitable marine environment in hundreds of millions of years. Understanding why this mattered requires understanding the concept of ecological carrying capacity. Every environment can support only a certain number of organisms, determined by the availability of resources: food, nutrients, space, light. All of these factors limit how many individuals can survive in a given area. When a population reaches the carrying capacity of its environment, growth stops, competition intensifies, organisms struggle for the limited resources available. During the early boring billion, marine life was pressed against the carrying capacity of the available shallow shelf habitat. The total extent of oxygenated, sunlit, relatively nutrient-rich water was limited. Every organism that could live in these environments was already there. There was no room for expansion, no opportunity for new forms to establish themselves without displacing existing ones. The fragmentation changed this equation. By doubling the shelf area, it doubled the carrying capacity. Suddenly there was room for populations to grow. Organisms could disperse into new territories without immediately encountering resource competition. And in these new territories, local conditions might be slightly different. Different water depths, different current patterns, different proximities to river mouths, creating opportunities for local adaptation. The expanded shallows were not uniform. Some shelf regions were broad and gently sloping, with extensive areas of very shallow water. Others were narrower, dropping off more steeply into the deep ocean. Some were adjacent to river deltas, receiving regular inputs of fresh water and nutrients. Others were far from major river systems, more nutrient-poor but also more stable in salinity and chemistry. This environmental variation was crucial for diversification. In a completely uniform environment, there's little opportunity for different adaptations to succeed. But in a heterogeneous environment, different strategies can work in different places. Organisms adapted to high nutrient conditions can thrive near river mouths. Those adapted to oligotrophic conditions can succeed in the clearer waters far from land. Spatial heterogeneity promotes biological diversity. The Stromatolite Gardens expanded into these new territories. Different stromatolite morphologies appeared in different settings, reflecting local environmental conditions. In high-energy environments with strong wave action, stromatolites formed as robust columns that could withstand physical stress. In protected bays and lagoons, they grew as broad, flat mats. In intermediate environments, domical forms appeared. The fossil record from this period shows increased stromatolite diversity, with new forms appearing that hadn't been seen earlier in the boring billion. Microbial mat communities also diversified. The basic structure remained similar, layered communities of different bacterial types, each occupying a preferred depth within the mat, but the specific composition varied by location. Some mats were dominated by cyanobacteria, others had significant populations of purple sulfur bacteria. Still others hosted diverse communities of heterotrophs that consumed organic matter produced by the photosynthesizers. For eukaryotes, the expanded shallows were transformative. These organisms, with their higher metabolic demands and lower tolerance for environmental stress, benefited enormously from having more habitat available. Populations that had been confined to small refuges could now spread across broader areas. Genetic exchange between populations became easier as distances decreased, and the larger population sizes provided more raw material for evolution to work with. The fossil record shows an increase in eukaryotic diversity beginning around 1.4 billion years ago, shortly after the fragmentation began. New morphological forms appear, the size range of preserved cells expands, with larger eukaryotes becoming more common. This suggests that conditions were improving enough to support the higher energy demands of larger cells. Some of these eukaryotes may have been photosynthetic, containing chloroplasts acquired through endosymbiosis, similar to how mitochondria were acquired earlier. Photosynthetic eukaryotes could occupy similar niches to cyanobacteria, but their larger size and more complex cellular machinery gave them different capabilities. They could potentially store more nutrients internally, allowing them to survive through periods when nutrients were scarce. Other eukaryotes were likely heterotrophic, consuming bacteria or other eukaryotes for nutrition. These organisms filled a different ecological role, acting as predators that controlled bacterial populations. The presence of predatory eukaryotes may have actually promoted bacterial diversity, preventing any single bacterial type from dominating and creating opportunities for less competitive species to persist. The expanding shallows also created more ecotones, transition zones between different environments. Ecotones are often hotspots of biodiversity because they contain organisms from both adjacent environments, plus specialists adapted specifically to the transition zone. The boundaries between oxygenated and anoxic water. Between 1.4 billion and 800 million years ago, something subtle but significant occurred in Earth's oceans. After hundreds of millions of years of apparent stasis, eukaryotic life began to diversify. Not explosively, not dramatically, but steadily. New forms appeared in the fossil record, genetic lineages that would eventually give rise to all plants, animals, and fungi began to split apart. The foundations of modern biodiversity were being assembled. This diversification was enabled by the slightly improved environmental conditions of the later boring billion. The expansion of shelf habitat provided space. The modest increase in trace metal availability provided metabolic resources, and the accumulated innovations of the earlier constrained period, efficient metabolism, sexual reproduction, basic multicellularity, provided the biological tools necessary to exploit these opportunities. The fossil evidence for this diversification comes primarily from microfossils preserved in fine-grained sedimentary rocks. These fossils, often called acritarchs when their biological affinity is uncertain, show increased morphological diversity through this period. Early in the boring billion, preserved eukaryotic cells are simple, spherical, or slightly elongated, with smooth walls. Later specimens show more varied shapes, spines, processes, complex surface ornamentation. This morphological diversification reflects biological experimentation. Surface ornamentation might have served various functions. Spines could prevent cells from sinking too quickly, keeping them in the photic zone where light was available. They might have deterred predators, making cells more difficult to engulf. They could have increased surface area, enhancing nutrient uptake. Each variation represented a different strategy for survival in the nutrient-poor, low-oxygen ocean. Cell size also increased during this period. Larger cells have certain advantages. They can store more nutrients internally, providing reserves to draw on during lean times. They have lower surface area to volume ratios, which can reduce water loss and make them less vulnerable to osmotic stress. They're more difficult for predators to consume. The appearance of larger eukaryotic cells suggests that conditions were improving enough to support the higher metabolic costs that come with increased size. Some of these larger cells may have been among the first multicellular organisms. True multicellularity, where cells remain connected after division and differentiate into specialized types, is difficult to confirm from fossils. But some specimens from this period show chains or clusters of cells that appear to be connected, suggesting coordinated colonial or multicellular organization. The diversification wasn't limited to morphology. Molecular clock analyses, which used genetic differences between modern organisms to estimate when their lineages diverged, suggest that the major eukaryotic supergroups began to split apart during the later boring billion. The common ancestor of plants, the common ancestor of animals, the common ancestor of fungi, all of these lineages were differentiating during this time. These divergences weren't producing recognizable plants or animals yet. The organisms were still microscopic, still relatively simple, but the genetic machinery that would eventually enable plant photosynthesis, animal movement, fungal decomposition, all of this was being assembled. The crown group of eukaryotes, the clade that includes all living eukaryotic diversity, was taking shape. This genetic divergence was facilitated by sexual reproduction. By mixing genetic material from different individuals, sexual reproduction creates variation that natural selection can act upon. In a stable environment with no sexual reproduction, populations tend to converge on a single optimal adaptation. But with sexual reproduction, genetic diversity is constantly being regenerated, allowing populations to maintain variation and respond more quickly when conditions change. The slightly improved conditions of the later boring billion may have provided just enough environmental variation to make sexual reproduction advantageous. Local differences in oxygen availability, nutrient concentrations, and predation pressure created a patchwork of selective environments. Offspring that were genetically diverse had better chances of finding a suitable niche than genetically identical clones. Predation likely emerged as an important ecological interaction during this period. The first predatory eukaryotes were probably simple organisms that engulfed bacteria or smaller eukaryotes through phagocytosis, the process of surrounding and consuming a particle. These predators would have been size-limited. They could only consume prey smaller than themselves, but their presence would have created new selective pressures. Prey organisms evolved defenses, larger size, harder cell walls, spines, toxins. Any adaptation that made consumption more difficult would have been favored, and predators evolved counter-adaptations, better mechanisms for detecting prey, more efficient digestion, the ability to handle defended prey. This predator-prey arms race, operating over millions of years, could have driven substantial morphological and physiological diversification. The diversification also had ecological consequences. As eukaryotes became more diverse, they occupied more niches within the shallow marine ecosystems. This increased the complexity of food webs. Instead of simple linear chains, photosynthesizers being consumed by heterotrophs, there would have been more intricate networks with multiple trophic levels and lateral connections. However, this diversification remained constrained by the fundamental limitations of the boring billion environment. Oxygen was still low, limiting how large and active organisms could become. Nutrients were still scarce, limiting total productivity. The deep ocean was still euxinic, restricting life to shallow zones. The diversification was significant compared to the preceding stasis, but modest compared to what would come later. By 800 million years ago, near the end of the between 1.4 billion and 800 million years ago, something subtle but significant occurred in Earth's oceans. After hundreds of millions of years of apparent stasis, eukaryotic life began to diversify. Not explosively, not dramatically, but steadily. New forms appeared in the fossil record, genetic lineages that would eventually give rise to all plants, animals, and fungi began to split apart. The foundations of modern biodiversity were being assembled. This diversification was enabled by the slightly improved environmental conditions of the later boring billion. The expansion of shelf habitat provided space. The modest increase in trace metal availability provided metabolic resources, and the accumulated innovations of the earlier constrained period, efficient metabolism, sexual reproduction, basic multicellularity, provided the biological tools necessary to exploit these opportunities. The fossil evidence for this diversification comes primarily from microfossils preserved in fine-grained sedimentary rocks. These fossils, often called acritarchs when their biological affinity is uncertain, show increased morphological diversity through this period. Early in the boring billion, preserved eukaryotic cells are simple, spherical, or slightly elongated, with smooth walls. By 800 million years ago, as the boring billion drew to a close, life had achieved something remarkable. The marine biosphere, which had been so constrained and limited for so long, now contained all the fundamental components necessary for the next stage of evolution. The genetic toolkit was in place, the cellular machinery was refined, the ecological relationships had been established. Life was ready. Readiness is a strange concept to apply to evolution. Evolution has no foresight, no goals, no capacity to prepare intentionally for future conditions. Organisms adapt to their current environment, not to environments that might exist in the future. Yet in retrospect, it's clear that the innovations developed during the boring billion were precisely what was needed for life to exploit the environmental changes that would soon arrive. The eukaryotic cell, perfected over 1 billion years of constraint, was ready for higher oxygen levels. The mitochondria that had been acquired early in eukaryotic evolution could scale up their energy production if more oxygen became available. The respiratory machinery was already in place. It just needed more fuel. When oxygen levels finally rose during the Neoproterozoic oxidation event, eukaryotes were equipped to take advantage. Multicellularity, which had originated during the boring billion, was ready for nutrient enrichment. Building large bodies requires abundant resources. During the constrained period, multicellular organisms remained small because nutrients were too scarce to support anything larger. But the genetic and developmental mechanisms for coordinating multiple cells, for differentiating cell types, for organizing tissues, all of this had been worked out during the era of scarcity. When nutrients became more abundant, these mechanisms could be deployed to build larger, more complex bodies. Sexual reproduction, with its capacity to generate genetic variation, was ready for environmental instability. The boring billion had been stable, but the Neoproterozoic would be anything but. The planet was about to experience some of the most extreme climate fluctuations in its history. Populations with genetic diversity had better chances of surviving these fluctuations than genetically uniform populations. Sexual reproduction, refined during the stability of the boring billion, would prove essential for surviving the chaos ahead. The ecological relationships established during the late boring billion were ready for ecosystem complexity. Predator-prey dynamics, competition for resources, symbiotic associations, all of these interactions had been developing in the simple communities of the shallow shelves. When environmental conditions improved and permitted higher biodiversity, these ecological relationships could scale up, supporting more species and more complex food webs. Even the metabolic efficiency forced by nutrient scarcity would prove valuable. Organisms that had evolved to function with minimal trace metal requirements could outcompete less efficient organisms when resources became slightly more abundant. The adaptations developed under constraint provided competitive advantages when conditions improved. The readiness extended to specific capabilities that would become critical in the Neoproterozoic. Some eukaryotic lineages had developed the ability to form resting cysts, dormant stages that could survive unfavorable conditions. This capability would be essential during the global glaciations, when much of the ocean surface froze. Organisms that could enter dormancy and wait out the ice ages would survive. Those that couldn't would perish. Other lineages had developed mechanisms for coping with oxygen fluctuations. During the boring billion, oxygen levels were low but probably variable, with periodic anoxic events on the continental shelves. Organisms that could tolerate these fluctuations, that could switch between aerobic and anaerobic metabolism, or that could survive brief periods of anoxia, would be better equipped to handle the oxygen swings of the Neoproterozoic. The diversification of the late boring billion had also created a reservoir of genetic diversity. Multiple lineages existed, each with its own adaptations and capabilities. When environmental conditions changed, this diversity meant that at least some lineages would be pre-adapted to the new conditions. Mass extinctions would occur, but not total extinction. Some organisms would survive, carrying forward the innovations of the boring billion. This readiness wasn't obvious at the time. To an observer looking at the late boring billion ocean, life would still have appeared simple and limited. Microscopic organisms in shallow waters, microbial mats on the seafloor, nothing visible to the naked eye larger than a grain of sand. The ocean would have looked empty, barren compared to modern seas teeming with fish and marine mammals and seabirds. But at the cellular and genetic level, enormous complexity had been achieved. The cells floating in those shallow waters carried within them genomes that encoded sophisticated metabolic pathways, complex developmental programs, and refined regulatory systems. They possessed the molecular machinery necessary for building multicellular bodies, for sensing and responding to environmental cues, for reproducing sexually and generating genetic diversity. The boring billion had been a chrysalis, a protected period where fundamental transformations occurred out of sight. The constraints that defined this era, rather than merely limiting evolution, had forced it in productive directions. Scarcity had driven efficiency. Low oxygen had driven respiratory sophistication. Limited habitat had driven competition and ecological complexity. Every constraint had, in its own way, prepared life for what was to come. By the end of this period, life had answered all the basic engineering questions required for macroscopic complexity. How to coordinate multiple cells, how to differentiate cell types, how to transport nutrients within a body, how to reproduce in ways that generate variation, how to compete with other organisms, how to avoid predation, how to extract energy efficiently from limited resources. The solutions to all these problems had been worked out during the billion-year pause. What remained was for the environment to change in ways that would allow these solutions to be deployed at larger scales. And that change was imminent. The tectonic forces that had held the continents in stable configuration for so long were about to accelerate. The supercontinent Readiness, which had assembled during the later boring billion, was approaching the point of breakup. And that breakup would trigger cascading environmental changes that would finally break the chemical cage that had constrained life for so long. The readiness achieved by the end of the boring billion, then, was not an ending. It was a beginning. Life had completed its apprenticeship. It had learned the lessons taught by constraint, and it was poised, though it couldn't know this, to inherit a world that would soon offer opportunities undreamed of during the long, quiet billion. As the boring billion drew to a close around 800 million years ago, the planetary conditions that had defined this era began to shift. The changes were gradual at first, barely perceptible over millions of years, but they were building towards something profound. The tectonic stability that had persisted for so long was ending. The climate equilibrium was becoming unstable, and the chemical constraints that had limited life for 1 billion years were about to be shattered. The catalyst for these changes was the supercontinent Readiness. This massive landmass, which had assembled during the later stages of the boring billion, had reached its maximum configuration. The tectonic forces that had brought the continents together were now being overwhelmed by forces pulling them apart. Deep within the mantle, convection cells were reorganizing. Hot plumes of rock were rising beneath Readiness, pushing upward against the base of the continental crust. The supercontinent was doomed. The first signs of impending breakup appeared around 750 million years ago. Volcanic activity increased. Rifts began to open along zones of weakness in the continental interior. Earthquakes became more frequent. These were the precursors of what would become one of the most dramatic episodes of continental reorganization in Earth's history. But before that reorganization could complete, the planet would experience something even more extreme. The position of Readiness, unlike the earlier supercontinents of the boring billion, was shifting toward higher latitudes. Paleomagnetic data suggests that parts of Rodinia were moving into polar regions. This geographical shift had profound consequences for climate. When continents are positioned at the poles, they can accumulate ice, and ice, with its high reflectivity, can trigger powerful climate feedbacks. As Rodinia began to break apart, the resulting increase in volcanic activity released carbon dioxide into the atmosphere. But the enhanced weathering of freshly exposed rock surfaces, created by the rifting process, consumed carbon dioxide even faster. The balance between volcanic outgassing and weathering, which had remained stable during most of the boring billion, was tilting toward a net removal of greenhouse gases from the atmosphere. The climate began to cool, slowly at first, then with accelerating pace. Ice accumulated at the poles and began to spread toward the equator. This ice expansion increased the planet's albedo, its reflectivity, causing more sunlight to be reflected back into space rather than absorbed by the surface. This cooling created more ice formation in a runaway feedback loop. The planet was sliding toward a global glaciation. Between roughly 720 and 635 million years ago, Earth experienced at least 2, possibly 3 episodes of extreme glaciation, collectively known as the Cryogenian glaciations or snowball Earth events. During these episodes, geological evidence suggests that ice may have extended all the way to the equator, potentially covering the entire ocean surface. Life was pushed to the brink of extinction, but the organisms that survived these ice ages were the ones that had been refined during the boring billion. The eukaryotes, with their efficient metabolism, their ability to form protective cysts, their genetic diversity created by sexual reproduction. These were the survivors, and when the ice eventually melted, as it did after each glaciation, these organisms found themselves in a transformed world. The melting of the ice triggered massive episodes of continental weathering. Meltwater carved channels through freshly exposed rock, pulverizing minerals and carrying enormous quantities of nutrients into the ocean. The nutrient famine that had characterized the boring billion ended abruptly. The ocean was suddenly enriched with phosphorus, iron, and trace metals on a scale not seen for over 1 billion years. At the same time, atmospheric oxygen levels began to rise. This increase, known as the Neoproterozoic oxidation event, brought oxygen concentrations from less than 1% to perhaps 5 to 10% of modern atmospheric levels. This still wasn't high by modern standards, but it was an enormous increase compared to the boring billion plateau. For the first time in 1 billion years, there was enough oxygen to support active, mobile, macroscopic organisms. The combination of nutrient enrichment and oxygen increase created conditions unlike anything in the previous billion years. Life, which had been refined and ready, could finally exploit its accumulated innovations. Multicellular organisms grew larger. The first macroscopic eukaryotes appeared, and by the Ediacaran period, starting around 635 million years ago, complex soft-bodied organisms were leaving impressions in seafloor sediments. Visible life had arrived. The storm that ended the boring billion was both destructive and creative. The Cryogenian glaciations nearly extinguished life, but they also created the conditions necessary for complex life to flourish. The extreme weathering during deglaciation ended the nutrient famine. The tectonic reorganization of continents created new habitats. The oxygen increase provided metabolic fuel. Every constraint that had defined the boring billion was lifted or dramatically reduced. By the time the Cambrian period began, around 540 million years ago, the transformation was complete. The ocean hosted animals with hard shells, active predators, complex ecosystems with multiple trophic levels. Life had explosively diversified in ways that would have been impossible during the boring billion. The Cambrian explosion, as this diversification is called, was the delayed result of innovations developed 1 billion years earlier. The approaching storm then marked the end of one chapter and the beginning of another. The boring billion, with its constraints and stability and slow refinement of cellular machinery, gave way to the dynamic, volatile, and ultimately more productive world of the Neoproterozoic and Phanerozoic. The billion-year preparation met the environmental opportunity, and life finally released from its chemical cage, seized that opportunity with extraordinary speed. The quiet billion had taught life patience. It had forced innovation. It had prepared the biological foundations for complexity. And when the storm arrived, bringing chaos and catastrophe and ultimately opportunity, life was ready to respond. The stage had been set. The actors had been trained. And now, as environmental conditions transformed, the greatest show in Earth's history was about to begin. There is something quietly profound about the boring billion, a span of time so vast that human minds can barely hold it, yet when examined closely it reveals itself not as a failure of evolution, but as its most patient achievement. For 1 billion years, the Earth held life in a state of gentle constraint, not killing it, not allowing it to flourish, simply maintaining conditions that demanded efficiency, innovation, and endurance. The constraints were everywhere, in the sulfidic depths that poisoned the ocean, in the vanishing trace metals stripped away by chemical reactions, in the slow erosion of flat continents that barely delivered nutrients to the sea, in the oxygen ceiling that refused to rise. Each of these limitations could have been a dead end, a barrier that evolution could not overcome. Instead, they became teachers. Life learned to do more with less, to build complex cellular machinery from scarce resources, to generate genetic diversity through sexual reproduction, to coordinate multiple cells into cooperative bodies, to survive in marginal environments where conditions were barely adequate. Every innovation that would later enable the explosion of visible, complex life was refined during this era of scarcity. The boring billion was preparation, not intentional, not guided, but effective nonetheless. When the planet finally changed, when continents fragmented and nutrients flowed and oxygen rose, life was equipped to respond. The billion years of patience paid off in the dramatic diversifications of the Neoproterozoic and Cambrian. But those later explosions were only possible because the foundation had been laid during the quiet time. Perhaps there is something to learn from this about time itself, about how the longest periods of apparent stillness can be, in reality, times of deep transformation, about how constraint can be as powerful a driver of innovation as opportunity, about how the universe prepares its systems, slowly and without fanfare, for the changes that will eventually come. The Stromatolite Gardens of the boring billion are gone now, their builders long since replaced by more complex organisms. The sulfidic oceans have been oxygenated. The flat continents have risen into mountains and eroded away again many times over. But the innovations developed during that quiet billion persist. Every plant, every animal, every fungus alive today carries within its cells the machinery perfected during Earth's Middle Age. The boring billion was not boring, it was foundational, and in the deep history of our planet, it stands as a reminder that sometimes the most important changes happen slowly, quietly, beneath the surface, in times when nothing seems to be happening at all. Thank you for walking through this vast stretch of time with me. May your own quiet moments prove as generative as Earth's great pause,